Assalamu alaikum, Dr. Atya. So we're just starting.
Wassalam.
Okay. So, Bismillahirrahmanirrahim. Assalamu alaikum and welcome back, ladies and gentlemen. So we are beginning with the technical session, the first technical session of day 2 of our international course on outbreak investigation and control of infectious diseases. Our invited speaker, the very first invited speaker for the first technical session is Professor Dr. Atiyatul Wahab. Professor Dr. Atiyatul Wahab is a full-time professor at Dr. Panjwani Center for Molecular Medicine and Drug Research at International Center for Chemical and Biological Sciences, University of Karachi. The title of her talk today is Repurposing of US FDA-approved drugs against NS5 from dengue virus and SARS-CoV-2 main proteases, MPRO, by NMR spectroscopy. Just to, before I request Dr. Atiya to begin her talk, I would like to introduce her in a bit detail. Professor Dr. Atiya is working at Dr. Panjwani Center for Molecular Medicine at ICCBS, University of Karachi, since 2010. She has completed her PhD in organic chemistry in 2007 at HEJ Research Institute of Chemistry. Afterwards, she undertook her postdoctoral research in structural biology under the supervision of a Nobel laureate, Dr. Kurt, at Scripps Research Institute, USA, and followed by a postdoc at Goethe University, Germany. She has 86 research publications and 6 US patents and 1 book and 2 edited monographs on her credit. She has also supervised and trained 37 foreign and 72 Pakistani scholars in the field of bioorganic chemistry. She has been awarded with Productive Scientist from the PCST, which is Pakistan Council for Science and Technology, in 2018 and 2015 at the national level. Professor Dr. Atiya is leading NMR user group and access to scientific instrument program in the country. With this, I would request Professor Dr. Atiya to please begin her talk, and would also like to thank her for time for giving this talk on behalf of comstech. Thank you.
Thank you so much, Fazima, for very kind introduction. And definitely it's a very prestigious opportunity for all of us to learn from our Iranian delegate. And I'm very happy and pleased to share some of my research with them and other students and colleagues. So today I will be discussing about repurposing of US FDA approved drugs against non-structural protein 5 from dengue virus and main proteases from SARS-CoV-2 by using NMR spectroscopy. Can you see my slides? Can you see my slides?
Yes, ma'am.
Oh, okay, thank you. So all we understand that drug repurposing is an approach in which we can identify new indications of already approved drugs, and that's something very exciting and that actually help us to bring some information or contribute as a developing world in the drug development process. Since the development of drug require billions of dollars and a huge number of scientists in different areas of research, and about 12 to 15 years time. So this approach is very exciting because we can find out different indications of already approved drugs, and there are many drugs which are in the market. They are actually developed for some other purpose and now being used for the treatment of some other diseases. And it actually saves lots of time. And being a developing world, we then able to contribute somehow in the drug development process. So the first topic in which I will be discussing is repurposing of drugs against main proteases of SARS-CoV-2. So as we know, the topic of SARS-CoV-2 and COVID-19, we all know very well. And there are main protease. There are actually a couple of proteases, PL-Pro and M-Pro. So these two proteases are very important, which actually help the viral and play important role in the replication of viruses. And this M-Pro is being conserved in corona virus family, and there are many computational X-ray crystallography approaches being used for ligand protein interactions, and many structures have been deposited in the protein data bank. Here I'm discussing about an important method, which is NMR-based method to identify those molecules that are interacting with M-Pro. And these M-Pro, here I would like to explain that when after the entry of the virus particle in the cells, M-Pro and PL-Pro, these are two proteases which play an important role in the proteolysis. And after this proteolysis, many non-structural proteins and other cellular replication of virus started. So if we inhibit or target these two proteases and inhibit them, then it means that we can stop the replication of virus. So I would like to explain STD NMR, which is saturation transfer difference NMR spectroscopy. It's an important technique, and it's I would say like a medium throughput technique that we have been using nowadays. We have like on resonance and off resonance spectrum. So off resonance spectrum are those spectrum in which all the resonances of protein and ligands we can see. So this NMR experiments is actually ligand-based NMR experiments. So the concentration of the protein that we choose is usually very low, most often between 2 to 5 micromolar concentration of the protein, while the concentration of the ligand that we use is 100 or 200 folds more than the concentration of protein. So if we are using 1 micromolar protein, then 1 millimolar of the ligand are used. So in the beginning, there are many optimization steps that need to be done. Since the concentration of the protein is very low, so usually we don't, and in the pulse sequence, there are the suppression of the protein signals sequence are also there. So we don't observe the resonances of protein signals, but we observe only the resonances of ligands. So these sharp signals are actually represent the signal of ligands that are interacting with the protein. So if the protein is large enough, so it means when we give, we need to saturate a protein, means we give a short burst of energy, a selective pulse, usually in those regions which are far from the signals of ligands, at least 1 to 2 ppm away from the signals of ligand, because we would like to make sure that we are not saturating ligands itself. So we saturate or give a pulse to usually upfield region of leucine and lysines, which are resonating below 0 ppm in the proton NMR spectrum. So usually -1 or -2 ppm. And when we give a short burst of energy, then since protein is a very big molecule, so energy immediately transfer to the whole protein and through a spin diffusion process. And then definitely when ligand bind to the protein, then it's a ligand and protein complex with each other. So during this complex procedure, so the energy which the pulse that we have given is transferred to the ligand of the signals of ligand. And here the signal of the ligand is usually enhanced. So only those part of the ligand which are interacting with the protein got saturation from protein. And since it has many limitations, so those ligand which are tightly bound with the protein, and then we cannot get the STD NMR spectrum. Since this is a dynamic process that one ligand get energy or saturation from the protein, and then it's a reversible process, so then other ligand occupy the place and get the saturation. So that is why we are using a higher concentration of the ligand. So maximum number of the ligand get saturation from protein, and we can get better signal to noise ratio. So here you could see that in this cartoonic example, this is off resonance spectra. Off resonance is like a proton NMR spectrum. So here in the on resonance spectrum, in which we are saturating protein, and the energy is transferred to ligand. And then we subtract on resonance spectra from off resonance spectra, and we can get the difference spectra. That's what the name shows: saturation, transfer, difference. So we got the difference spectra, and this difference spectra is the one shows only those protons which are interacting with the ligand. And here, in this example, you can see that in this there are signals of ligand, and when we get STD difference spectra, then means saturation and on resonance and off subtracted on resonance and off resonance spectra, then we get the difference spectra. So here you can see only these protons which appear here from this one are the one who are receiving saturation from protein, and the other protons are away from the protein site of binding. So in this way, we can easily understand the transfer of saturation from protein to ligand, and it doesn't require any kind of labeling. So it's a very easy and very easy method, and it's also very cost effective. So we use this technique to identify those ligand which are interacting with protein. So this is again the similar type of the example that I have shown uh, to you. So we have evaluated 155 drugs and a few compounds um, using STD-NMR spectroscopy with the AMPRO enzyme. And the enzyme um, we have purified and expressed in our own lab. So actually, we have discussed this when we wanted to use STD-NMR spectroscopy for the screening of drugs. Then we discussed with Professor Iqbal Chaudhry, who's the Coordinator General Comstech. He is very renowned scientist. He suggested and proposed us that instead of screening each individual drug, we need to make mixture of the drugs. And then we should have like quick and dirty screening. But so we try and make a mixture of 5 drugs. And in this way, we have screened 35 mixtures in the first step. And these mixtures have also it may have that there are some interactions that we may observe due to interaction of many ligands together. But the experiment that we use is kind of short experiment, the same experiment, but less number of scans is being used. So usually it takes about 30 or 35 minutes to finish one experiment in STD-NMR of mixture. But then we carefully evaluate, since we already have the proton NMR spectrum of each individual drug, we carefully compare each proton NMR spectrum with the STD NMR spectrum of each mixture. And then we can find out, okay, these are the drugs which are present in this mixture shows some kind of interaction with AMPRO enzyme. And then we run the individual experiment with one drug and protein. So out of 155 drugs, we have identified 10 drugs that showed some kind of interaction with AMPRO enzyme. And here I would like to explain that my colleague, Dr. Sabah Farooq, we have asked her to evaluate these 10 drugs against SARS-CoV-2 assays, which is antiviral assay, plaque reduction assay. So she has done these experiments, and so we screened in the first go 155 drugs using quick and dirty method of an STD-NMR spectroscopy, and then those which we have identified as 10 potential drugs that shows interaction with AMPRO. Then we have side by side, we ask Dr. Sabah to check the antiviral assay, and we also check this individual drug using NMR spectroscopy. So these are the plaque reduction assay that Dr. Sabah has performed and confirmed by real-time PCR. So here I would just a minute. Okay, here I would like to show the chloroquine phosphate, which is being using as an antiviral drug. And when we have done as a standard, we want to take some standard also. So this drug is reported for anti-malarial and herbicides and widely used for the prevention of treatment and prevention of malaria. So this is the proton NMR, the on-resonance NMR off-resonance NMR spectrum, and this is the difference NMR spectrum. So we could see that these colors actually shows the relative percentage of saturation. Since we cannot identify the real percentage of saturation, the highest the saturation is received is being designated as 100%, and the other are actually related to those 100% proton. So these percentages are a related percentage of saturation. And here we could find out that these two proton signals shows 100% saturation, and with the yellow one was 50%, and the green one 28%, and blue one with the 16%. And then we could also find out that in the plaque reduction assay, which shows that 82.3 micromolar concentration, 100% reduction, and 41.5 micromolar, 63.2% reduction. So this is the same way that we have identified all our inhibitors. And here another drug, which is remdesivir, which is a broad spectrum antiviral drug and is being used for SARS-CoV-2 as well. So we could find out the interaction of these protons, since it's the NMR is a technique in which we can easily identify at the molecular level or at atomic level that which proton is or which hydrogen is interacting with protein. And so here we can also using antiviral assay, we can see that it shows 82.3 micromolar concentration, 86.7 micromolar percentage reduction of the plaque. The next is dexamethasone, which is a corticosteroid and used for to reduce the allergic symptoms. And here we could find this out that in STD NMR experiments, these are the protons. The pink one, it shows the highest interaction, and which is 100%, and then yellow one 58%, and then the green one 54%, and the blue one is 51%. So these are the relevant percentage of saturation they receive, and with the plaque reduction assay shows 77.2% reduction. So here is another drug, which is diatesium hydrochloride, which is a calcium channel blockers, and we could easily find out using STD NMR experiments that this methyl showing 100% interaction receive 100% saturation, and these are the methyl receive 31%, and we could see only these two signals there. So, and 81.8 percentage inhibition, percentage reduction. And we also perform docking studies, and which represent that, because proton STD NMR shows which part of the ligand is interacting, but then the docking, we would like to predict which part or which amino acid is being involved to with the interaction of these small molecules. So and here we also done the simulation studies, which shows that the ligand and protein complex is fairly stable. and more stable complex means it can work in better way. So another drug, which is mefenamic acid, also shows interaction with the with the AMPRO of SARS-CoV-2, and this is non-steroidal anti-inflammatory drug. And here we could see that these two metals are interacting, and we can see there is no other signals in the difference NMR spectrum. And using plaque reduction assay, it shows 81.8% reduction. And the molecular docking and simulation studies shows different type of hydrogen bonding, aromatic hydrogen bonding, pi-pi interaction. And here is the 50 nanosecond simulation, shows the protein ligand complex is fairly stable. And similar is with the other drugs, which is lost and potassium. And the same study we are presenting here. So I just pass all these. So another drug is mexiletine hydrochloride, which also shows interaction with the with AMPRO, and here is a docking studies result, which shows different type of hydrogen bonding and pi-pi stacking. And here initially it's like a bit not unstable, but then it becomes the complex become more stable. And another drug, which is amantadine hydrochloride, which shows interaction. The whole almost just a small molecules, almost all protons are interacting, and 77.2% reduction. And the docking studies and simulation shows the protein ligand complex is fairly stable. And then we also checked the effect on the stability of the protein in the presence of ligand. So here all these 10 molecules is being evaluated. This is the TM, the melting point for the protein, which is 54.5 degree Celsius. And we could see that these two, which is glucine hydrobromide and trimethylbutane malate, are the one who actually reduce the TM quite a large level, from 54 to 52 degree Celsius. So which shows that, and other also shows some are stabilizing the molecule, but these are the ones which actually reduce the TM, the melting point. And the next, I would like to present about drug repurposing against dengue virus. Dengue is a problem in our area. So we have selected non-structural protein 5 from dengue virus. And this protein has two important domain, which is AMPase domain and RNA dependent RNA polymerase domain. So we actually have a screen on the whole domain of the whole protein, which has both domain for the using STD-NMR experiments. And this is the same experiment that we have used, and we have evaluated 75 FDA-approved drugs. And among these drugs, they are grouped in 15 mixtures, and 10 were identified as a lead candidate to be further study. So here is just an example I would like to show. That is a diclofenac sodium, which is interacting. Here is a proton NMR spectrum and the difference NMR spectrum. So again, the relative percentage of saturation with the red color shows 100%, and the blue one shows 57% of saturation. The yellow one shows 36% of saturation. The green one shows 7.8% of saturation. and the orange shows 19% of saturation. So these are the proton that shows different percentage of saturation that receive from NS5 of dengue virus. So we also done the docking and simulation studies for this, and to identify which amino acids are interacting with the ligands, and which type of the interactions are there. So and here is the same study that we have performed, like itopride, and that shows again interaction at different interaction of different protons using STD-NMR. And so these are the atenolol and the different drugs that we have identified for the study. And then after identifying using an STD-NMR experiment, we have performed inhibition assay, and we of all these 10 molecules using NS5, and we could find this out that on the one which diclofenac sodium is actually the one which are which is stopping which inhibiting the polymerase reaction of NS5. So he and at the end, I would like to say that we using these molecules and NMR experiments, we could identify those drugs that shows interaction that has, and to using the drug repurposing approach. And I would like to acknowledge my students Abdul Mateen and Ismatullah for their work, and Dr. Humaira Zafar for helping in docking studies, and the financial support of the Cell Company, HEC, Pakistan Academy of Sciences. So thank you.
Can you hold me? Okay, so, yeah, thank you very much for validating an extremely important science. In fact, we have developed this brand new method for identification of very rapid antiviral drug discovery method by using biophysical technique of anti-magnetic spectroscopy. I was wondering whether we can use it for the whole virus, instead of expressing protein. Virus can be crystallized, there are many X-ray substances supported. Can we do that? Because here the size of virus should not be better. The only thing we need to do is to saturate virus molecules, virus molecular assembly, and then see which molecule is in close proximity or form interaction with virus. Can also be theoretically used for viral detection, detection of which virus or which family exists. So just a wild idea may not be practical, but just wanted to have your views about that.
Thank you so much, sir. It's a very nice idea. I will work on it and then discuss with you further.
If anybody from the audience has any questions?
Thank you very much, Chair. This is Kiran Zorbanet. I'm not very familiar with the technique of NMR, but I have also a few questions. First of all, in your diagram, the x-axis is ppm, and I don't know what the ppm is in this context. So the
I'm sorry, I can't hear you well.
No. So the scale of the x-axis is ppm. I don't know what ppm is in this context. And another question, I assume that you have expressed the protein in E. coli in the purified air. And just like chicken and so other groups also have done the same. So I wonder if any post-translation or modification is important for this enzyme or not. So in that case, you may need to either purchase the mammalian cell produced protein or do it by yourself, difficult. And in your graphs, all of them, you measure the flame reduction at a certain concentration of 82.70 micromolar. I think this was the concentration that you found for the first drug. So have you used the same concentration for other drugs, or this is the or this is just for the first drug, and then you have tested the interaction of other drugs with this drug, and then you realize that, for example, the drug reaction has decreased to 50%, 70%, and so on. And just a comment about what said, I think it's not possible for this virus because this is an input virus, so it's not easy to purify it and crystallize it intact. I think that this is done for adenovirus, for example, for the virus which are protein in structure. So, but I have not seen this for envelopes which are covered with the typical structure. Thank you very much. I think there's a echo device. You want me to repeat the question of Brother Kehan Ramesh? You want me to repeat his questions?
Yes, sir. Sir, I couldn't
Okay, so
understand.
the first question was in your NMR spectrum, the x-axis was ppm. So can you tell us what this exactly means on x-axis, which is, chemical state?
Oh, okay. Sir, let me answer first one. So this NMR is actually time domain. The real spectrum is in time domain spectrum, and using Fourier transformation, we convert into frequency domain spectrum. So these are, and then frequency, if we use only frequency for each spectrometer, it would be different. So because if we record the same spectrum on 500 and then same molecule on 800, then the resonance frequency will be different of each nuclei. And that is why we multiply with the spectrometer frequency, and we divide with the spectrometer frequency and then get it in the parts per million, so that the chemical shift remain the same. Am I right, sir?
Can I translate your response?
Sir, please.
Okay, so what he's saying is that the x-axis is hertz. Okay, so it is a frequency scale. So don't worry about ppm. ppm is just to, because the equipments can be different. So you just try to normalize it against the standard. So the first question and the second question was the expression, why you need to express it in E. coli? Why can't you do it in mammalian cells?
Or it is not important to have, isn't it is there any post-translation or modification on protein which requires the mammalian expression?
You heard him, right? Okay.
So he's absolutely right. Those protein which has the post-translational modification that need to be expressed in mammalian cells. Since I this other than this, but expression in mammalian cell is not as high as in E. coli, and secondly, since my basic area of research is structural elucidation of the protein, so we need very high concentration of the protein, at least 1 millimolar concentration of protein, which we can get, and we need to label the protein as well. So that's why we established this it's like more friendly and easy to do using E. coli system as compared to the mammalian. But those which require post-translational modification that need to be expressed in mammalian cell is absolutely right.
And then he commented on my proposal of using viruses as macromolecules for radiofrequency transmission through nuclear over other effect to the attached molecule as a tool to identify and the interacting molecules, disrupting molecules, as well as biomarkers for. So, you being the only structural chemist, structural biologist in the country right now based on NMR, you need to prove me wrong based on his hypothesis, okay?
Okay, sir.
I have another comment also. These proteins are not present in the virus itself. So they are just produced during the replication cycle inside the cell.
Okay,
So that not that.
so this is a very valid comment, but I remember that you have conducted long time ago an experiment in cell.
Yes, sir. Yes, sir. Yes, sir. We have done this.
Those
Yeah.
proteins which are present, can be done, I think, in cell without expressing them, right?
Yes, sir, definitely we can do it. And the only concern in those experiments, we have to be really fast because the cells usually settle down with time, and it will be out of, NMR. There will be in the tube, there is a range that the observer, the detector detect the signals. So that's the cells usually settle down. So we need to be fast in experiments. There is no other problem with those.
Thank you very much, Professor Satya. Professor Satya was trained by Professor Woodbridge, who is a Nobel laureate.
Anyone from the audience who wants to ask a question? Okay. Thank you very much, Professor Athiya, for your very informative and wonderful presentation. So we move on to the next session, which is environmental investigation, and it will be conducted by Professor Dr. Arash Arashkia, who is the Assistant Professor of Medical Biotechnology and Head of the Virology Department at the Pasteur Institute of Iran, and Dr. Fahmi Amiri, who is the Assistant Professor of Epidemiology and member of the Research Center for Emerging and Re-emerging Infectious Diseases at the Pasteur Institute of Iran.
Hello, good morning. Welcome to second day. Hello, good morning. Welcome to second day of course. In this section, I'm going to speak about environmental investigation. Actually, environmental investigation should be closely integrated with epidemiology and laboratory investigation. And if the epidemiological investigation are available, it should use for focusing on environmental investigation. And if the agent is known, we need to consult with laboratory investigators for know about the transmission routes and focus on the places who we need to do the environmental investigation. Depending on the type of outbreak, the environmental investigation can help form a hypothesis. And it is good for immediately address to control of the outbreak. But before going to do the investigation in environment, you need do some basically actions. For example, you need gather background information. You need to become familiar with type of process that are likely to be encountered and regulatory environments and standards for this process. At the first, you need to determine which agency has been legislative authority to investigate. Sometimes you need to hand this part of investigation to this agency, and sometimes you need to conduct a joint investigation. You need to review some guidelines that are important in these sectors. For example, if the outbreak is related to food and it's foodborne, you need to review the hazard analysis, critical control point, HACCP system, and food safety programs. Or if the outbreak is related to hospital and maybe in institutional outbreaks, you need to review the infection control guidelines. Sometimes outbreaks are related to pools. So there are some guidelines for pool management. Sometimes the outbreaks is related to drinking water. So you need to review the treatment plant operation guidelines. Before going to site visit, you need to identify and make contact with the key individuals that are involved to the outbreak. You need to make some relationships and with the persons who are responsible for the outbreak. And during the initial discussion, it is recommended that when you are going to talk with the key individuals, you should present the basic details about the outbreak frankly and openly. And if the source of the outbreak has not been identified yet, you should clearly state that. If the epidemiological analysis is not complete yet, you shouldn't present any suspicions about the outbreak sources. Then you need to arrange an acceptable time for the site visit with that individual that are responsible for the outbreak. Then identify whether there are any forthcoming events, if which the circumstances of the event under the investigation may recur. If the outbreak could be associated with the human source, for example, carriers and infected food handlers, identify the number of potential sources and how they may best be contacted. After doing these basic actions, you need to conduct a site visit and inspection. Doing this act provides the interface between the investigation and conduct an outbreak. And it is likely to have maximum benefit that undertake this action undertake as soon as possible after identification of the suspect site. And when you are when you do observation during the site visit, it may reveal helpful for some clues about the outbreak source. It may address general hygiene and safety issues, and it can directly lead to implementations of control measures, regardless of the subsequent epidemiological findings. Sometimes we need to do the site visit at a prompt visit and early, because we need to identify sampling and stop or remove from some events that are related to the recur of the disease. For example, stop sale or any food that are that may be contaminated. And but sometimes we need to do some secondary visits because we need more information about the event. But keep in mind.
Can I ask you a question?
Yes, sure.
You see, these are outbreaks which are very common, like diarrheal outbreaks. Or in case of a mysterious outbreak, like the one in Wuhan or Ebola in Ivory Coast, how would the epidemiologists would react to it? Because the nature of the bug is not known. How would they handle it? And to whom they would report? Because this is something which they don't have.
In case of environmental investigation, about the propagated outbreaks like COVID-19, it's recommended that doing the environmental investigation is not very necessary, and it's not helpful because we can't find the resources of in environment. But sometimes we need to do some basic investigations for found the resources. For example, in COVID case, Dr. Arash will say about some studies that yes, the challenges of the corona in the China for finding the reservoirs. But the environmental investigations are important in other cases of the outbreak, not propagated sometimes. I will talk about these situations in next slides. Thank you.
For this specific question of what we should do or what they should do in Wuhan, Arash has a very good explanation of what they have done. And they have done a great job, I think. It is not conclusive, but it's very well done. And this session is dedicated just to the environmental study. So we have other aspects about the case contact, the clinical steps, which are not covered in this specific session. But we have discussed it and we will discuss it when we come. Thank you. Thank you.
You're welcome. In site visit, just keep in mind you need to 3 components: place, procedures, and people. In doing investigation in place, you should keep your mind open and gain a general impression. And because sometimes unpredicted factors may related to the outbreak, and it can appear apparent when you keep your mind open. You should identify any past event or situations that may have contributed, and consider whether specimens of leftover in point outbreaks, for example, foodborne diseases, is associated with the common event, and collect specimens immediately, and because when we are going to a site visit, we need choose the specimens immediately, but when there are a lot of risk factors and we don't know anything about the outbreak, and we are we have to select specimens about the agents, about the, for example, every surfaces, transmission route, out risk factors. So maybe you need to select some more samples, but it is recommended to just store the specimens until you get some information about the outbreak. And after know anything, a little clue about the outbreak, you can do tests for the specimens. It is important that you be cautious about the widespread testing in our environmental specimens, because sometimes routine environmental culturing usually lead to results that cannot be interpreted, and many surfaces and areas have some normal environment and are contaminated with some agents that are not related to our outbreak. So they can take us in wrong way. The next component is investigating the process. Closely, you should observe the process and procedures, including those the conditions that are normal or not worth to mention. And if it, you need to overlook some, you need to just investigate some process such as storage, distribution, and other things that are important in the processes. Just look for gross negligence and contamination or gaps of techniques, and check whether policies, protocols, and manuals are available to use. For example, HACCP or the management guidelines for pools, and then check against the current standard, and review monitoring and recording keep systems that are obtained specific monitoring data, reveal the that place. Sometimes outbreaks are related to humans. When in these situations, you need to identify a list of all individuals who may have come into contact with the suspected outbreak source, and interview each individual in a standard questionnaire, collect specimens if it's appropriate. For example, when cook or handlers are suspected for some foodborne outbreaks. Last day, we talk about some outbreak patterns that were for point prevalence, point outbreak, continuous outbreak, intermittent, and propagated. Now, today, I'm going to speak about more detailed outbreaks based on the exposure on time and place. So we have some common events and that are outbreaks due to exposure of a group of persons are exposed to agent on same time and same place. Like the point outbreaks, and it's the example for this is like weddings, like some parties, and others. So in this case, when you are faced with an point outbreak, you should go to site visit and inspection. You need to review the HACCP-based food safety assessment and public health risk management plan-based water assessment if the water is suspected. Collect of food, water, and environmental specimens, and collect of clinical specimens. For example, maybe you need to fecal specimens from the food handlers. We discussed about this study yesterday. It is an outbreak related to Bacillus cereus in iftar occasion in Indonesia. In this case, the environmental investigation for assessing this outbreak, they interviewed 2 food handlers to get information related to number ordered for food for iftar and to evaluate food safety procedures, including the process of cooking, storing, serving, and etc. In addition, they used a checklist for assess the kitchen sanitation. And they collected stools samples and food samples from the ceremony. Other type of the outbreak is common source in a specific place. In this type of outbreak, the outbreak is has common source, and it occurs in same place but in different time. Anybody can give me an example for this type of outbreak? The same place but the different time.
Water source?
What?
Water source and diarrhea.
Water source?
And diarrhea.
Malaria. Seasonal malaria.
Malaria? No. No.
The COVID sources. It's not a common source.
The place is same, but not the same time. People who visit this place, yeah, for example, in restaurant, for example, pool, or some place like farms. People when visit there, they can get ill, but not the same time. And the time is not same for all the people. So based on the source of the outbreak, for example, if it is waterborne, you need to visit in swimming pools, review records of use, fecal accident, colorization, staff illness, and protocols. If the case is about zoonotic disease and it's for farm visiting, you need visit potential sources and review level of human exposure, and also the some hygiene system in the farm. If the outbreak is foodborne, you can act like the common event. So for foodborne, you can site visit and inspection. You should review the HACCP based on food safety assessment, or if it's related to water, you need to public health risk assessment plan based water assessment. Then you need to collect of food, water, and environmental specimens, and collect clinical specimens if it's possible. For example, There is a study on pool-related outbreak of Cryptosporidium that they found an outbreak cryptosporidiosis among users of 2 swimming pools. In this case, the investigators didn't take any samples from the pools, but they just reviewed the processes. For example, they reviewed the pool policies, management, and operating procedures, pool water treatment, routine microbiological sampling results, pre-swimming hygiene practice, and pool records. They just reviewed that and consult with a chemical expert to found that the hygiene chemical are great or not. Other type of outbreak is dispersed common source. These outbreaks are due to exposure of a group of persons in a community to an agent that is common to the individual in a group where exposure have not all occurred around the same place, necessarily around the same time. Some examples for this type of outbreak is drinking water system or some contaminated food that produced. In these cases, based on the outbreak, you need to do some actions. You need to act as common events when potential source identified, and site visit and site is different in this case. For example, it's manufacturers or food processors or hospital, or maybe the place is different here. You need to review the water treatment sanitation and review the records if the water is source of the outbreak. Other type of outbreak is community-wide or propagated outbreaks. In propagated outbreaks, as I mentioned to Dr. Iqbal, it is not environmental investigations is not important sometimes, but the propagated disease are sometimes complicated. For example, CCHF has different ways for transmission. It has the person-to-person transmission in hospitals. It has vector-borne transmission in the in outside, and it has zoonotic transmission in the farm, for example. So for this type of the outbreaks, we need, if we want to investigate overally, maybe we need to take some do some investigation in environment based on the ticks or vectors or animals. For example, these are our activities on pasture in this one. They are collecting the ticks from the farms by using a plan. And here they are investigating the animals, and they collected some rodents from the wild animal for investigating the some infections. Sometimes outbreaks confined to population of a specific residential or other institutional setting, like hospitals or rest home or prisons or schools. So in these cases, we need to site visit, environmental risk assessment, review the HACCP-based food safety assessment if it is connected with a food, and collection of the environmental specimens. When the outbreak is related to airborne microorganisms, you need to check the ventilation system too in the institute. And the last type of outbreak is household, and it's confined to members of single household. Household outbreaks probably occur frequently, but are likely to be underreported. Sometimes in some outbreaks of vaccine preventable disease may be linked to a batch of vaccine that has been inactivated by incorrect storage or handling. So environmental investigation in this type of the outbreaks are important. You need to check the process and check the cold chain maybe. And that's it. An interesting discussion remained by Dr. Arash, and he will talking about the COVID-19, and it is very interesting discussion.
Okay.
Yes. Any question? Yes.
So I have this. For example, if we investigate the environment samples, for example, in case of AMR, and they are the more sources. For example, there are environmental sources and animal sources that transfer disease from animal on environment to humans. And it's already all investigation has decided, so okay, these are the two source of gene transmission AMR. So what will be further steps to like conclusion for the finalized transmission of the final source of like AMR genes from animal, either in animal or environmental to human. Yes.
Okay.
About that. But last example that I about it can be confirmed, but with a great program and wide program about considering the human environment and the animals, and it is one health approach, and it's important for some issues like AMR. I think you need to do environmental study in AMR issues and integrate with animals and human studies. If can I answer you or
Well, I think this question is very complicated.
Yeah.
You have to talk about, or I just wait for Arash's presentation, because he also will explain the complicated.
Yeah.
Then we can come back and say.
Yeah. Any other questions?
Okay, please come.
Sorry.
Oh, it's okay. Thank you for doing that. Okay, this is Arash. from Pastor Institute of Iran. It's my great pleasure to be here with you, and I'm going to talk about the source of SARS coronavirus, especially its challenges, because it has not been determined. Firstly, to show you that how the situation was for SARS, tracing back the original viral diseases may take decades. It's what in the textbooks they say. Masked palm civets, this animal. So this is, sorry to find it. Oh. This animal, masked palm civet, has been the intermediate host in the Guangdong, China for SARS virus. And this picture was taken by me from the Natural History Museum of Islamabad on Saturday, that you have the same animal too, because it's the other name is Himalaya palm civet. But yours here, I think it's jangly belly, I think in order it has written. It's not masked, it is only palm civet, maybe he has not used proper PPE, so it's not masked. But since SARS was epidemic in 2002 to 2003, it took about 15 years to find out that the bat was the source, was the exact source. See how for SARS coronavirus it could be. Even the original Ebola virus, as well as those of many other viruses, have not yet been determined. You know, for viral diseases, we are not sure even finding the source is really necessary or somehow a little bit helpful. But in any case, we are going to talk about the scenario for SARS-CoV-2 more. You know what happened exactly, and we are going to talk based on the published data. I'm not going to judge the labs, the China government. I'm just going to talk about the published data that are now in the public domain. There were samplings from this market, Hunan Seafood Wholesale Market, located in Wuhan, China. That is thought to be one scenario is that this market was the place that SARS coronavirus was jumped from the animal to the human. They took samples from the environment. I mean, we are going to talk about environmental sampling, and found genetic materials from different animals that were Amur hedgehogs, Malayan porcupines, masked palm civets, Siberian bisons, and other rats and other animals, along with the viral genetic material. But among the present animals, just raccoon dogs, that are kind of foxes, they are really, were susceptible to SARS coronavirus too. None of them were susceptible. So it became the suspected case. And they say now that raccoon dogs may be the main, at least intermediate host for the SARS coronavirus too. Okay, now we are going to see the criteria or evidences that not really their criteria, they are evidences that people have posed to find the exact scenario. Coming to left below, in China, raccoon dogs are commonly sold for their meat and fur. Okay, they are present in China in Wuhan market to us. Raccoon dogs and other wild animals were at the market in Wuhan, China at the time that SARS coronavirus became epidemic. We are going to just see how the evidences could help or could pose more challenges. SARS coronavirus 2 was found in many of those same market stalls. I mean, overlapping genetic material of animals and the virus at the same spot. It was found. Raccoon dogs can be infected with and transmit SARS coronavirus 2. These are seems strong evidences. And finding a susceptible animal at the same place around the same time that the first people caught COVID. So these somehow strong evidences may suggest, may point to scenario which the virus has jumped from the animal to the human. And now more evidences are coming. What are they? That market, that Wuhan market, has two wings, two buildings, in west building and east building, divided by a road, by an avenue. And most of the virus positive samples were clustered in the market's southwestern corner. These are found by Western, most of them by Western researchers. In the same place where start selling live animals were previously found, because the market is divided to different places. In one place, they sell live animals. Of course, it's now closed. Half a dozen virus positive samples were also positive for raccoon.DNA or RNA. It's against pro to the scenario of the raccoon duck being the intermediate, at least, host, along with the human genetic material also. And this way, the market could be an early epicenter of SARS-CoV-2. So so far we have very good evidences to say that the market was the source for SARS coronavirus 2. To find out better, researchers should go back to upstream of the market, to the wildlife or the farm that the animal have been bred, at least to find some progenitors of the virus. That should be done for the future. The same was done for the SARS, that the people came to the bats after 15 years. So maybe something like this should be done for SARS coronavirus 2. Okay, now we have some evidences pro to the market being the source. Now there are some critics posing some very strong comments. What are they? Just to show you how environmental investigation could be so complicated, so troublesome. There were animals in the same exact spot with and without virus. So one thing that is posed some challenges. Much human DNA presence in the samples that were taken from the market, and large amounts of viral DNA have been found in fish and seafood. We know that they are not the host, they could not be, but even the viral load, I mean, the amount of genetic material of the virus in the seafood and fish was even higher than the raccoon duck. And it poses a great challenge to the raccoon ducks to be the intermediate host. So, two scenarios have been posed by the critics. The previous evidences that we named them were circumstantial evidence, might be. And the worst case scenario, it was a lab leak. We will talk about it more.
So what is the source of human DNA? Was like handling or?
Yes, they thought environmental, from the environment. I mean, from the stalls, from the doors, and they found both human DNA and viral DNA, and also the animals one. And more challenges are coming to show you how complicated it could be. What it is, if the market was not the place that SARS-CoV-2 crossed from animal to the human, but instead the site of a super spreader that people were already infected, with the people that were already infected. Why there? Why exactly in the same market? The second one, if human brought it here, to the market I mean, why did they bring it to the place in Wuhan with the most stalls selling wild animals? Okay, it's very challenging to answer these questions.
Like criminal investigation.
Yes, exactly, because American security agencies are doing the same, because these are the questions that have been posed by intelligence services. To say that recently the 7 security agencies in USA gave their reports to the president. 4 of them came with the result that with low confidence it has been come from an animal. 2 of them with low intermediate result that it has come from the laboratory, and 1 not decided. So it's very, even the kind of investigation didn't come with the conclusion. WIV, that I was there about 1 month ago, Wuhan Institute of Virology that is accused for releasing the virus, is the most credible virus research center in whole China. So every other virology center in China for reference laboratory go to the WIV. And they have a lot of experience working with coronaviruses. They are working since SARS-1. The guy that Professor Shi Zhengli, a very nice guy there, who talked about risk management with us, has been in BSL-4 in France since 2002, or PhD.
But they have their BSL for us.
Yes. And now they have two, but one doesn't work exactly. And the point is that 1 year before the epidemic or the pandemic really, they established their BSL-4.
By French.
Yes, exactly. And they are 10 miles away from that market. It's not so near. I thought that it is very near to the market, but it was 10 miles away, and now it's close also. One more point that in an article in Science has been published by all the American scientists from Scripps Research Institute, I think. They found two lineages of the virus, lineage A and lineage B, that had two genetic mutations, very few mutations regarding an RNA virus. But you may hardly find it here, that they found lineage A and lineage B in two different distances from the market. They found lineage B, one case, these are the first cases, that was their genome was released by the Chinese CDC. One lineage A was around 1.2 kilometers away, lineage B, sorry. Another one was 8 kilometers away, while lineage A was 2 kilometers away. And this way, through some statistical analysis, they say that they were two lineages coming from the same market. It couldn't be a human that goes twice to the same market. And because critics pose this comment that possibly one has gotten the virus and mutated twice inside and then spreaded it. They come to this conclusion, it's somehow impossible that one person goes twice into the market and gives his virus to the market and say, this way it has spread it.
So lineages and variants are different.
Just two. That's why the critics could pose this criticism. The mutations is very, regarding an RNA virus, they are very low. The critics.
Just one comment. This terminology about the classification of viruses is very flexible. So when sometimes very few mutations come to isolates, to lineage, to mutants, things like this, they're changeable. But what's his point is that these two strains have just different in one point mutation.
2 point mutation.
But anyway, they are distinct.
Yes, you can find distinction.
But they existed at the same time.
Yes, in different, say, distances from the market. The people who has got them, because these are human samples. But for critics, this is very strong, say, support for the critics, and very great challenge for the supporters of the big market at this, the source. I mean, it is a timeline challenge. Here you can find that the samples from the market that we talked up to here about them, the evidences, were taken on about January 2020. The first confirmed COVID cases were around December 10, 1 month earlier. Even virus possibly began circulating even as early as mid-November, 2 months earlier. So after 2 months, these documents have been collected, and those evidences that we talked about so far were based on those evidences. 2 months earlier, it could be the virus might be spreading in the market or in the, say, community. By the way, Possible scenarios based on the what we talked about are in public domain, they say. A raccoon, dog, or other intermediate animal has directly infected a human in Wuhan or elsewhere, and then the people came to the Wuhan. A bat directly infected a person outside Wuhan, who then brought the virus back to the city, because the bats that carry similar viruses aren't found in Wuhan. A scientist.
They aren't found.
Yes, the bats aren't found. A scientist from one of the Wuhan Institute of Virology labs got infected by a bat while doing field work because they were doing it. A scientist at one of the labs collected the virus sample from a bat or other animal, brought the sample back to Wuhan, and became infected while working with it in the lab. And the worst case scenario is the gain of function studied in the lab. You may know what are gain of function studies. They change the genes of a pathogen or any microorganism to say how it affects that microorganism. Sometimes it may come to the conclusion of that, for example, that microorganism could infect human cells easily, more easily, I mean. And they are accused for such a study because they were doing gain of function studies on SARS coronavirus, but there's no evidence for it really.
But is this a common practice of conducting such studies?
No, it should be BSL-4, and especially for the airborne viruses, it is very risky. It's highly risky and should be done in the most containment laboratories, because it could pose a very serious danger.
Serious biosafety.
Yes, exactly. Yes, exactly. It's very risky. By the way, finding sources may help, may not. But in an outbreak, the al-hazard approach is needed. You may not come to the conclusion that you need to know the source, but you do your environmental investigations to find what is happening really. It might be a human outbreak, disease outbreak. It could affect anything: water, health, human, soil, feed, public health, or critical infrastructure, everything. And it could be human disease outbreak, biological attack, bioterrorism, other environmental events, even cyber attacks, natural disaster, chemical attacks, chemical and oil releases, and nuclear happenings. So environmental investigation is needed, but it may not come to the finding the source of the outbreak or any other disaster that may occur. That's all with me. I will be with you for biosafety session afternoon. If any question or
Japanese
yes.
recently reported in a very credible journal. I'm trying to remember that. I'll give you the. They said that the early COVID-19 samples, both from environment and human, contain various variants which were later claimed in 2021 and 2022 late. So they claim that these variants were already there in samples. What do you think about that?
It's another accusation of Chinese that they say if you had the.
It was a Japanese paper.
Yes, I know.
I have it. Actually.
They have been accused for not releasing the data until last year, until 2022, because they were telling that they were not telling they have the samples, the environmental samples. But the Chinese are really very conservative now, and they try to keep the information a little bit with themselves, to make situations calm down. As far as I understood, I could feel really, because people didn't talk about it. We were there for biosafety course, and we were not in the position to ask them, who did you do really? And we were not the people to ask them. But what I could feel is that they were very conservative to give the data, because they were telling just in the first session, they tell that everything we speak about especially USA presidents, General Secretary General, or other security agencies find something to blame us. So they were even thinking of how to release data. Maybe in the future they can find. Even they have recently published a literature, an article about the source, and even they didn't claim that the laboratory is not the source. They don't say it. They try to scientifically go through the evidences to say that it might be. They say it might be the market. Even they themselves are very reluctant, are very conservative in releasing the data. It's understandable completely because Zheng Li, she was really upset about the situations. She was telling us that it was not my right to be like this. We are all scientists. And it was bad for all scientists to blame them that you can do something that may pose danger for all people in the world.
But this is sure that human-animal interactions has actually brought many varieties.
Exactly. The same for SARS, for Ebola, for Marburg. No, yes, no. This is a very good example for them, but there's because of the gain function studies that they were doing at the same time and timeline challenges. Really, the critics are very strong now to blame them, and giving the clue is very, very hard, very difficult. Yes.
Another example, they tried then plus.
Yes, exactly.
Spread was reported in like maybe maybe should collect some claim or accuse the United States they have developed this virus. But now with the newer techniques, especially with the molecular genomic studies, genomic surveys, and we talk a little bit about genomic surveillance now. It is clear that there have been multiple rounds of transmission between human and monkey and different kind of apes. So they went back and forth, and then now this is what we have.
Even I
You know,
write a paper,
Yes.
I given you reference, so it's now, there are many questions regarding the HIV is basically is not zoonotic. So like, for example, there is more events like less than only few cases just reported. So on this basis, we do not claim that it's zoonotic. I will give you a paper and reference, so it's mentioned clearly that.
The main problem in this case was being pandemic, because it became pandemic, many people died because the one student, Kenya student that was working in the Dr. Zheng Li lab was telling me that in those days, everybody calls laboratory and they tell everything they wanted. They tell that you killed my family, everything in that time. That's why the situation goes so hot topic, because they had SARS, they had Ebola, but very limited number of cases and even more mortality rate. But the number of cases were low, so it was not so hot topic that days. This virus is somehow different.
So I question, corona is also present in animal.
Yes. Every animal has its coronavirus.
That there are just evaluate from further dangerous species, and then it's evaluate like COVID.
It could be like influenza. It's not. It's the genome structure is different because influenza could be somehow drift and shift, but here it is not the same. But if you go to the molecular replication of the coronavirus, you will be amazed how it goes for replication and transcriptions. And the jump from the animal, because when I was working on MERS in 2014, I think, I was going for detection. I could find many statements in the discussion of the articles that they were telling, this is not the last coronavirus. Maybe the next one becomes pandemic. I can find them even now, the same sentence. And when it happens, I said, oh, it came. Because it was predicted. But they say that we don't know when, we don't know to what extent. And it happened in a good extent, but fortunately the mortality rate was really low because it could change the population of the whole world.
So the many studies of viruses jumping from animal sources to Nordic.
Yes.
Yes.
Human. I think the vice versa is also taking place from human to animals.
At least for, because we are not going for them in the animals, but it is possible.
Somebody,
Theoretically, it is possible.
so there may be a lot of extinctions.
Maybe,
Animals.
maybe.
Maybe because of that, human viruses transferred to animals. But theoretically, it is
Yes.
equally possible.
We don't care for animals, but it could be
No, no,
theoretically.
we do.
Yes. No, I mean, they're viruses. I mean, not animals by themselves.
That's in my opening address, I mentioned that these viruses are replicating and they evolve, and they have a role in evolution and life on the earth.
Yes.
But the problem is that when some of them get too pathogen. So otherwise we can bear with them. They are here to be here.
So I remember in one of the quotes of Sapiens, and Harari has written that many of these large animals in Caribbean and South Pacific islands disappear because human viruses affected large mammals. They all went out. You don't find them anymore.
Mhm. Very nice. Yes, oh, please, sorry, he had the first, sorry. Okay.
But the common concept of Chinese people was this, that we are like dealing with these wild animals, and we are there are some viruses which can pass to humans,
Yes.
but it's not very easy. There is a barrier, usually a barrier between. It's not very easy for a virus to just transmit to humans. And the Chinese people are saying that like thousands of years we have been eating, we are dealing with these wild animals again and again. So why is it so that at the specific moment it just came to humans and spread like fire? The second thing is that the people I'm not going to start a political talk, but there were Wuhan military games in November in Wuhan. Like after that, they just exploded. So, common people were talking on this one as well, that it can be bioterrorism, maybe.
You know,
Can be.
the problem that you said first for the old and ancient, I mean, happenings, really we are not aware of. Maybe in the, say, some restricted areas, something has happened, many people have died, and we are not aware of it even. Even not died, some of them died, like the SARS, what happened for SARS. Many affected people, but around 1% mortality rate. If it happens and no documentation, you can't find it. What happened for SARS? It was in 2002, 2003. Not a few, but several people were affected. The mortality rate was about 8,000. Yes. About 10%. Yes, it was a high fatality rate. For MERS, it was about 30%. But the number of people who get the infection were very low. Because they could not transmit it easily. This virus, its main advantage for itself, the main advantage was spreading very easily and affecting people. So this debate is a little bit is not supported really, because if you have a virus that jumps this way and could transmit easily, it can happen about 5 years ago again with another coronavirus or even any other viruses. About the political status, I'm not really aware of. What I saw in the in China, yes, they are military-based country, and they had the experience of SARS. So if they even suspected to something is happening, it was really, for me, it's logical to do something military to keep people. They quarantine people, although they at the first day it was very difficult for them, but if they have released their data correctly, their mortality rate is low.
Although we have passed time, but just to explain what she said, 2 months before that, there was an international game for military game.
Mm-hmm.
Teams were there, even from Iran.
Yes.
But she mentioned that maybe it was a terrorist act at that time. Because people from all around the world were there.
I, my personal belief when I was there in WI was that Dr. Zheng, she was here because regarding the previous outbreaks that happened there, because the Guangdong is not a very far province from there that SARS, the first SARS came to the public. So it could be because the climate is changing. There, because of the scarcity, they bring the live animals together. They keep it for a long time to sell them. They can jump the coronavirus because each animal has its coronavirus, and they can be recombinated to be a new virus even.
Even it could be considered, sorry, it's considered that already in August and July, the COVID similar like clinical have found in England. So what do you?
Okay, we are running out of time. Just to answer
No, his question is remaining.
If it is possible,
Yeah.
okay. But just to answer this question, we tested all the samples that we had such a suspension in Iran before the first case, and we couldn't find any more cases. Maybe the earliest one was something like 2 or 3 weeks before the COVID one. So we tested those cases, cases of influenza, but they were negative for influenza. Then we tested them again for COVID, and that was not the case. But the clinical picture is very similar to many other viral diseases.
Sorry, your question, the last one, please.
Actually, the SARS outbreak is not the only one we have already gone through it, but this was the second outbreak which is SARS-CoV-2. Why are they not sampling from the wild animals when you are dealing with live animals, and why are you not sampling for SARS in the live animals? Why they don't do this?
They have controlled it. It didn't need it really, because SARS at the time was really disappeared. There was no SARS because they couldn't find the vaccine against it because at the time RNA vaccines were not developed yet. But for SARS, they have no problem to continue. For MERS, even if it goes crazy, really things would be very dangerous. But still, WHO goes for some meetings, so for some endemic investigations. That's all they are doing for the MERS. Even MERS still exists, and annually they have some reports. SARS has been disappeared, but no one was believing that something pandemic would happen because it's a huge number of people, huge number of animals to be tested, and it required very great, say, logistic to do so. But maybe from now on, maybe they do. Okay, thank you, guys.
Thank you very much. So before we proceed for tea break, we have a small ceremony for certificate distribution to the honorable speakers. Just stay seated for a minute, please.
Thank you. Thank you.
So I would like to request you to kindly present the certificates to the honorable speakers. Thank you. Dr. Kihan, Asad Manish.
And thank you very much. Thank you very much. My municipal prize is made of gold and it has the logo. I was remembering this.
Dr. Arash?
Thank you very much. Thank you.
Dr. Baba?
Give them.
Dr. Hazmi?
Faham means wisdom, right? Thank you so much. So the spelling has to be.
Okay. Mr. Saeed?
Please receive your certificate for your presence in this event.
Thank you very much, everyone. You may join us for tea now. Thank you.
Okay, thank you very much. I'm waiting for a boss to tell me when to start. Hmm?
We're also waiting for.
Okay. Shall I start, boss? Okay. Bismillah ar-Rahman ar-Rahim. Now, this is the situation that I'd like to talk about this part. Well, actually, it's not like right now. Well, I learned it a lot because I have a lot of experience and product on this. And it is on laboratory investigation. What is the role of the lab in laboratory investigation, and how are the tests done interpreted, and which tests are more used? This is the subject that we would like to touch upon them. Maybe we are not able to discuss them in very much detail, but I hope that you will enjoy and it will be helpful for you. As in yesterday, I told you that if you are in any specialty that you are, you need to communicate with other specialties. So that's a critical, this coordination is very critical in outbreak investigation and also control. So you must understand what they need. Lab people should understand what they need beyond just one. Both of you should know what the medical doctors want. All of the logistic guys should know what you want. Okay, so this is a matter of communication. It's a matter of understanding, common understanding. And I want to start this with all due respect to Dr. Eshtati. Don't trust an epidemiologist in a suit. Okay, sorry. I mean, not any epidemiologist, but a field epidemiologist. When you go to the field, you should do their job. So it must be direct property. Something like this. This is famous husband. But books is not also an epidemiologist, is a microbiologist. But you see, Sana is behind me. You have everybody, and I think she was dressed in a cover suit for the field study. Of course, we did this. Or when we do lab studies, we do not have fancy labs in the field. So do whatever you can in the field. And this is an epidemiologist, the same guy that I told you yesterday, who is a veterinary doctor and who interviewed the veterinary. Can you see the way that he traced this mouse? It's very much like the another photo that I showed you yesterday, our late colleague, Mr. Hanifi, taught us how to collect fleas from mice. And this is what she does now. Okay. So what is the role of the lab, or what are the roles of the lab? Because based on the role that you want, because the expectation, the way that they work and the way that we interpret the result, the way that they interpret the results are different. So sometimes you want them to confirm the clinical diagnosis among a list of differential diagnoses. So first of all, the doctor sees the patient, he or she has a series of differential diagnoses and then asks the help of lab to rule in some of them. Or providing tools for case finding among the target population. For doing mass screening, for example, finding for people who are asymptomatic or test positive. Or providing data on trends of different diseases and or clinical diagnosis. Yesterday, we discussed about how to find out that there is an outbreak. So you must have some standard trends. And these trends in lab are very important because it does not, it is not just the positive results that you report. Even the negative results, even when you have no result, but the admission, the number of admissions, the symptom of the case, or the type of samples that are admitted to the lab are also important. Sometimes when you have a lot of, for example, samples of diarrhea, what is your expectation? Something is happening there. And you in the lab understand this even before the authorities. because they wait for the result of the lab, but you see that something is wrong. Or when we use this to monitor COVID-19, of course, during the control measures, not right now. And finally, what we discussed today, we have to find causes of outbreaks. The approach of the labs are also different. Some of them are patient-oriented, some of them are public-oriented. Some of them, the main clients are clinicians, some of them, the main clients, the government or the health authority, like epidemiologists. Some of them are designed to find the patients or look for diseases, some of them are designed to look for the health. And for some of them, diagnostic value is important for especially for the health public health laboratory, the cost benefit, the cost efficiency of the test are more important. So in the public health, we do not go for fancy tests or fancy labs. We do what we can do for a broad spectrum of people with as low cost as possible. Well, and if you go back to the steps that was introduced by Sana and Fahime, those 8 steps or usually more steps. In an outbreak investigation, in at least 6 of them, lab has the direct input or direct interaction with them. First of all, to identify an outbreak, for example, an excess of report of certain agent, or to establish a diagnosis, or to develop a case definition. And we discussed about this case definition yesterday. Sometimes it is a part of case definition. Usually it is a part of case definition. So as you mentioned, Sam mentioned yesterday, for example, for case of COVID, you have a positive test, when we don't the clinical symptom, this is the positive case. This is the confirmed positive case. But if you do not have, it might be either suspected or what was the other? This is another. Probably, okay. And sometimes even a specific type of disease is important. When you have an outbreak of, for example, diarrhea, maybe checking if it is O100 E7 or H7, what is it? One of them is E. coli. They are not viruses, so I'm not interested in them. Is important that it is especially stereotype very important. So sometimes even at the level of typing, you need to rely on the lab. Then to develop a hypothesis, for example, on the basis of current knowledge, we know that strain A is prevalent source. So maybe this strain is responsible for this disease there. And also, when you develop a hypothesis, you need to confirm it. And again, that helps to do that. And one very important role is to evaluate control measures. When you implement some control measure, the outcome usually is monitored. One main indicator is if this measure has decreased the number of positive cases or not. So lab is very important. Well, and you deal with different types of samples, from human materials, blood, urine, the sample directly collected from the lesion, to animal materials, to environmental samples, which was discussed just a few minutes ago. And we have different methods for identifying, for helping others in the lab. Some of them are direct. You look for the agent itself, but sometimes it's not feasible, it's not easy. For example, it's not really easy to find the Mycobacterium tuberculosis in the brain in the case of encephalitis or meningitis, TB meningitis, for example. So how do you detect them? How do you do the diagnosis?
Biomarkers?
Okay.
Test.
Well, it doesn't matter if you're right or right. I just want to interaction. That's very good. Thank you very much. Yes, biomarkers is a good answer. And these are indirect methods. So they check the response of the host or the surrogate marker. So biomarkers are usually surrogate markers. they not exactly show you or tell you that this is the agent, but they are indicators that this is the agent. Or for the host response, all IgG, IgM that we measure are the host response against something. Maybe the disease is already present, maybe it has been a previous model of the disease or whatever. Well, for direct methods, there are a lot of methods. So sometimes you just watch the sample that you see, just microscopically. Or for others that we go through them one by one. Well, for microscopy, for example, the consistency of stool is very important for us. Is it solid? Is it diarrhea? Or is it water rice? So what does it tell you if it is like this? Is it just like water rice? Cholera. Who said cholera? Okay. Okay, very good. So sometimes just looking at the sample gives you a very important clue that what you're dealing with. Well, for blood, the color of the blood, for example, sometimes hyperlipidemia, you have hyperlipidemia, and hyperlipidemia, and the color of the serum is different. Or especially finding the parasite in the stool, or finding blood in the stool also. So it also gives you some differential diagnosis in doctors. Then you may use a light microscopy without any processing, just very light microscopy, again for looking for parasite or protozoa, again in stool exam or in skin lesion, for example. Then you may go for detection by staining. Some of them are non-specific, like ground staining. Some of them are more specific, like Williamson or mycobacteria or Sporidium. And some of them are very specific, usually by antibodies against the immunofluorescence or antibodies related to some use of antibodies. And this is the favorite of virologists. So using electron microscope, it is not, usually it's not the first thing that we do. But when we have such a picture, we are very, very glad. So what is this one? Well, that's an electron microscope. You do not take an electron microscope from bacteria.
Electron microscope you are using for virus.
Okay, that's a virus. That's a filovirus. Okay, maybe this one, because this one was the Outbreak of 2007, 2006. So it is an old virus. I think this is what is this emergent fever? The name is from a German city. What? No. This is the emerging fever virus. What is the name of the German city? Found first in the German city. Yes. Is it in Africa? They found it in. Okay. Harvard, exactly. Yeah.
It's the name of virus.
Yes, but I think they found it in a city in Germany.
This is not the name of disease. Right.
Yes, because the virus was found in the city of Harvard. Yeah. So this is Marvel, and you see these are small tiny particles. I think these are HIV regressing from the cell surface. So this is also very interesting for us. We sometimes rely on them. And then you have these rapid tests. Now you're familiar with them during COVID. For different diseases, now they're available. For influenza, we have antigen tests, we have antibody tests. So this is the modality of this one, what they really test for urine antigen test. And what is the advantage and disadvantage of this test? So let me go back to this one. What is the advantage and disadvantage of this one? Well, viruses in the same family may look alike, yes. But even for that, you may add some antibodies and coated with gold particle, and then you have some specific micrograph too. But more than that, they are very difficult, they're very expensive. You need a purified sample, Okay, it is not something that you do for every patient and in a day-by-day activity. For rapid tests, they are very cheap. Well, not very cheap, because we will come to this. It caused a scandal during COVID. But anyway, somehow they're cheaper, they're easier to use. And they're very fast. What are the disadvantages? Well, not near 100%. So that okay, the sensitivity might be low or specificity, but that's true for all this. So what else? This is the qualitative test, but more than that, it's very subjective. So it depends who is reading the test, who is interpreting it, and that's somehow difficult to standardize. Well, then we have the tests which rely on propagation of the agent. These include, for example, bacterial culture, in some cases viral cultures, and well, for parasite, it's rarely used for parasite, but anyway, it's most commonly used in bacteriology and sometimes in virology. Even when you have an unidentified virus, you make a culture on different cell lines or permissive, terminology for different viruses. And then they wait for some time, and then they look for the cytopathic effects that usually are well, not characteristic necessarily, but give a clue of what kind of virus can make such a difference for the change in a cell. And then you can isolate them. And what is the advantage of this series of tests? What is the advantage of this series of tests when you propagate the agent?
So, Outbreak has
increase the number of viruses.
Yes.
Okay, so what?
Because we are using cell lines.
Using the DNA sample.
Yeah, what's the point? What's the benefit for you?
I think so. DNA sample for PCR if so it creates.
Well, this is not really true for viruses, but for bacteria, you can do the antiviral test after that. So, well, yeah, we are talking about an outbreak investigation, so you do not have time to do this right now. But anyway, that's right. You can keep it, you can study it in the future. And what are the disadvantages?
Culturing golden test is also like for
Basically, we grow the virus itself.
Yeah, that's exactly the same. So the biosafety and biosecurity concern is the major concern for this, and with Arash. Okay, Arash will discuss about the biosafety and mostly biosafety in the afternoon. And that's one problem. The cost is another problem. It's time consuming. It needs very expert people and very specialized facility. So it's also not a common practice, especially for the case of virus. Of course, you have a BSL-3. Who is from NIH here? Okay. So you have a BSL-3, so you can do that. Well, another problem is the viability of the agent, especially this is true for bacteria. For example, for meningitis, for Neisseria meningitis, the bacterium is very sensitive to temperature. So if the transfer chain from the patient to the lab, the temperature, if it goes to the refrigerator, it will not grow. So there are some factors that affect this. There is the interpretation result. We cannot be 100% sure. Of course, never you can be 100% sure. But anyway, this is a big problem here also. But you can do the antimicrobial resistance. That's the typing or typing. Typing is also important. Usually, we need more of the agent to do the typing, and then storage of the strain for future studies, for reference, and yeah, discuss this. Then it comes to the molecular tests, which are now very common. And one problem is that sometimes they are too common. I mean, not necessarily. We can do many things just with the previous strategies. We do not need this expensive equipment. But anyway, some of them are so-called target amplification methods, which means that they amplify the target sequence in something. And some of them are signal amplification. They do not amplify the target itself, but when they find the target, they amplify the signal based on that target. Usually we use this, there are tests of this, we're not going to discuss it, this is not subject really. But the PCR and its variants, real-time PCR and all the variants of PCR are among these, and also isothermal methods. What are the isothermal methods? Everybody know PCR? I assume yes, yeah? Okay, so I don't need to explain. What about the isothermal methods? Anyone else?
Like we are using it for mention PCR.
No?
Using
Using what?
A specific.
Okay. Okay. I'll tell you. This is this series of techniques like NASBOD, like LAMP, things like this. They do not need the thermal cycle that you need for PCR for separating the strands and then joining them. They work in a stable temperature, usually for example 42. So for doing them, you do not need a PCR machine, just thermoglycan is enough, and they are very cheap. They have their cons and pros. So, some of them are rapid, some of them are fast, some of them need more, all of them almost need less equipment. Designing, some of them are more, labor intensive, more knowledge. You must do the validation more than normal tests. But anyway, they are available, and sometimes we use them. And the last one is the sequencing. And this is now, one of the best methods that we have for many things, especially for both for detection and for identification and classification. And a little bit we did talk about the application of sequencing in the following slides at the end of this session. Well, so you or people in the lab know what they are doing, okay? But you must know what it means for the people in public. So we have different parameters for a test, like validity, reproducibility, sensitivity, and specificity. And you understand, maybe you are familiar with these concepts. For example, the other tests evaluate the right parameter for what we are looking for. I gave you the example of meningitis and TB. Usually in bacterial meningitis, your sugar level is low. It means that it is consumed by bacterium. Okay, and this is an indirect indicator for bacterial meningitis. So remember that. So this is not a test for bacterial meningitis. This is just a clue for bacterial meningitis. So it does not detect itself the bacterium itself, but a viral culture, when you have bacterial culture, so it is conclusive. So the validity of this one is much more than this one in case of meningitis. Is the valid. And then does the test give the same result for the same case every time that you test it? This is the famous figure, and you have seen it several occasions, I assume. So what is ideal is this? So you have a test that targets the target, the right target, and always hits the target. But actually, you are somewhere in between. This is the worst case because sometimes it's true, sometimes it's not true, and not always true, not always not true. So you cannot rely on it. And there are tests that they are sometimes true, but the others that they are not true are also close to be true. So they are fairly maybe acceptable. And there are tests that they are very reproducible, but they are always wrong. So that's a bad case. But there might be some methods, for example, to recalibrate these tests to give the right result when you have this reliable test. So our goal is to have these tests. And you are familiar with sensitivity. What is sensitivity of a test? Okay, before going to this slide, how many of you are people in the lab? Okay, what is the sensitivity of a test? What is the sensitivity of, for example, PCR?
We take the maximum quantity.
The minimum quantity. Well, now we talked about accuracy before. Or I'm not talking about accuracy now, about the sensitivity. So when you ask someone in the lab if this test is sensitive or not, what he or she perceive from your question is that what is the minimum amount of reagent that this test can detect? Okay, this is called analytical sensitivity. Okay, this is important for us in the lab, but it's not important for the others. So what is important for the others is the epidemiological sensitivity. And what is it? Okay, so great, Rohan. Okay, go on, please.
The ability of a test to correctly identify the positive cases.
Positive cases, okay, that's. Is that right? I'm teaching this for 27 years and I'm not sure if, not every time that I'm not checking it again. But I think that's true positive. So when you have a series of patients, you have some of them true positive, some of them are false negative. So the probability of finding true positive among all of these. This is called sensitivity. So for testing this, you take, for example, 100 people with the disease and test them one by one, and then calculate how many of them are detected by your test. Okay. Or in the mathematical way, the probability of having positive test in the case of being infected. And this is a specificity. Okay. Let me say so what is the specificity? True negative cases. Means that if you take 100 people disease free, how many of them are detected negative by your test? Okay. And who is the epidemiologist here? Okay. And do you like the sensitivity and specificity or not for a test?
Yes.
Okay. Okay. Now answer this question. You know, HIV is a very nice example for teaching what the tests mean, because the tests developed for HIV are of highest sensitivity and specificity, I think, in the history of medicine, because a lot of work has been done on that. So you have a lot of tests for HIV with 99.5% sensitivity and 99.1% specificity. Okay. Then you are here, everybody who passes with the test. Then you find a positive test. What is the probability of really being infected? And you know, these percentages are real. You have tested with this quality. So what's your answer?
99.99%.
Let the epidemiologist answer the question. So what's your perception? How much it should be? High, low?
No.
Why low?
Hi.
Why high? Okay, let's see. Well, for answer this question, the question is not the sensitivity or the specificity. It is the question of what is the probability of being infected in the case that you have a positive result. Okay, and this is the important factor for a clinician. So a doctor doesn't care about sensitivity or specificity of the test. He wants to know how much he can rely or she can rely on a given paper result from the lab to decide what to do with his or her patient. Okay, this is called positive predictive value. And what is it? It depends on sensitivity, specificity, and also prevalence of the disease. And that's the critical one. Okay. We have a parallel concept of negative predictive value, which is important for ruling for the diagnosis for a doctor. Okay, let's see. I will show you the calculations for another for the case of COVID-19. Just now I go to the results. If you have a test at specifically at 99.1%, okay, with the same sensitivity that we thought. If your prevalence is something like 0.1%, the positive predictive value is not more than 10%. Okay? You will see this why in case of COVID-19, but just take it for granted now. And then if you test in a population which, for example, 10% prevalence, this result will be 92% reliable. So have you ever asked yourself why we use different tests for HIV, for comparing HIV? So we first of all we do a rapid test or an ELISA test, then we go, for example, for Western blot or PCR. So what is the rationale behind it? Because ELISA is not good, you can greatly find the test with 99.5% sensitivity and 99.5% specificity in whole history of it. So the hits are perfect. So why don't you rely on their results?
Qualitative way.
No, that's the point. Okay, louder, please.
I think those virus then genetic material and
So what about genetic material? What's wrong with it? Okay, no, they did not change. So, okay, they changed, but not that good. The point is that when you do the first test, you pick someone from the population with a prevalence of 4.1% and place him or her in a population with a prevalence of 10% based on this calculation. So the second test that you do is checking someone in a population with a prevalence of 10%. So if you use another test, for example, this one, with this one, okay, this one is better. Or don't read this, not so much better. Now you are testing in a population with a prevalence of 10%. So now your result is 97% reliable. Okay, doing this serial test, although they test different aspects of the disease, so they are somehow confirmatory to each other. But the epidemiologic importance is that the first test, the screening test, isolates someone from a low prevalence population, put him or her in a high prevalence population, then the second test easily can detect. Okay, this is true for clinical symptoms too. If you do that, if my question was doing an ELISA or HIV in a population with say lymphadenopathy, with fever, with chronic fever, for example. So you know that prevalence of disease in this patient are higher than normal. Okay, so the prevalence of the original population is not something like this. It might be something like, for example, 5%, but people of this could be infected. So the value of your test would be something like this. And that's why in the case definition, we combine clinical symptoms or sometimes even the epidemiological, for example, if he has come from this certain area, if he or she has been in contact with the people with the infection. So they increase the value of the test. And that's why, you know, as a medical doctor, I was trying not to treat the test result, treat the patient. I do not rely on the test result to give my advice. So we must see the patient, people in the lab must know the symptom, the origin, things like this of the patient, then they can interpret the results. Okay, they do not have a magic test, something that to say that this is patient or this is not. Okay. In case of HIV, there's another also problem that this test detects different substances, different material. Most of the ELISA test or oral ELISA test take antibody. Some of them take also the antigen, the fourth generation test. And the video period is different for this. So there might be some cases that they are still negative for the antibody, but they are infected and they are positive for the antigen. So having a better test improves the accuracy of what you are doing. And I have very good examples of this. We can talk about it if you want. Well, we talked that why in case of HIV. In case of HIV, it's also an extreme case, but the stigma is very important in this case. So if you say someone is HIV negative, you prevent him or her from many things around normal people, normal person, okay? So for this, we are very sensitive to label someone as HIV positive. This was true also for COVID in the beginning. Okay, another aspect of this is, we use HIV for different purposes. For example, for epidemiologic surveys, for diagnostic, or for blood transfusion. What is the major concern when you do an HIV test for blood transfusion? What do you hope? Why do you test the sample for blood transfusion? Test HIV. I mean, what is your purpose? What is your goal? What makes you happy? What results make you happy?
Negative.
Okay, negative. That's right. Okay. So if you have any suspicion that this sample is positive, you just discard it. Okay. In this case, usually one test is enough for discarding the sample. Because if you have any suspicion, you discard it, you take it, work with the rest. So in such a case, maybe even one test is enough. For diagnosis, we discussed maybe 2 tests or 3 tests is required. For epidemiology surveys, it's usually in between. So mostly we rely on 2 tests. And also, in the past, in 1997, something like that, WHO had a very complicated guideline for how many tests to be used in different settings. And it also involved the prevalence of the disease on that city. At that time, some countries in Africa had a prevalence of something like 40%, 50%. Or in some special group, for example, there are groups in Iran, we had a percentage of 40.5, so high. So the interpretation and the requirements of the testing were different based on the population that you wanted to test. Okay. Finally, the most important limiting factor for testing, especially in the public health setting, is the cost. You may have 2 tests. One is excellent, gold standard, but expensive. You cannot roll it out to the population when you are in shortage of resources and can spend your resources, for example, for a scheme meal for the children. So you prefer not to go for the best results, but for the best you can afford. And it's very dependent on many factors: prevalence of disease, specificity of the test, cost of implementation. If you need to send someone to collect samples to do the test, if you have the technical facility, technical people who can do that, etc. Well, and what are the sources of error in the lab? Well, a lot. Just like what I said about the statistical test, don't be fanatic about our results. We are not so sure about our results too. So some of them are pre-analytic, which happen before doing the test. For example, during collecting sample, during labeling. For case of COVID, there is a time that if you collect the sample very, very carefully, the upper respiratory tract is negative. Okay. So for collecting sample, for labeling sample, that's a very common error. Then it comes to the testing procedure, instruments, kits, then the internal characteristic of the test. Tests that are reliable for 80%. So do not over rely on that test. And most of them can be also both or either random or systematic. And which one is better?
Random.
Why random? Why systematic?
Random provides more quality in different diversities and then population.
Okay, and you're not a person from lab.
Yes.
Okay, you're not, right?
Yes.
Okay. Are you from lab or not? Okay, lab prepared. Which one do you prefer to encounter in lab? And why? Why randomized system?
Because we have pre-analytic, analytic, and post-analytic. So we should focus on pre-analytic, analytic, and post-analytic.
No, the point is that if you find the systemic error, it's very easy to correct it. So just change your instrument. Just change the packaging way of the person. Just change the key. So you can find the source very easily, but for random errors, it's very difficult. So us in the lab, we prefer to face systemic errors. And of course, to find them. If they're not found, that's a disaster. Okay, and all of these factors can, actually affect the specificity and sensitivity of this. Usually what is written in the brochure of the kids is what they have measured in a very controlled setting in the lab. But when it comes to samples collected from periphery, from somewhere else, doing the test with the everyday laser system, for example, the results are not the same. So bear in mind that they are different. Well, I want to tell you an experience of pneumonia outbreak in April 2011 in Iran, which was unidentified. And it was characterized by lab, almost exclusively by lab, to see the power of what we can do. On that day, we received the reports that there is a respiratory outbreak in a barrack, in a military barrack in Isfahan, in central Iran. And people had cough and fever, I think, fever, and sputum, and one of them died by that. So the first differential diagnosis were flu, tuberculosis, mycoplasma, and pneumonic plague. Well, due to logistics, we received the samples midnight the next day, and we went to the place that they were separating samples in the National Laboratory Reference Health Reference Laboratory, and we started doing. They started doing a bacterial culture, and we did the rapid test that we had for plague, and they were negative. The next day, we did PCR for real-time PCR for all of these agents, and they all terminated. So what? So the next day, new differential diagnoses were suggested, and it was for adenovirus. So we did real-time PCR for adenovirus, and 3 cases were positive. And do you know what adenovirus causes usually in human? Adenovirus. But what is it cause? Something like common cold or conjunctivitis. Usually it's very light disease, so it doesn't make people hospitalized. So that was strange. And by that time, the first results of culture were reported, and they were, I think 3 of them, or some of them were positive for mycoplasma, for Streptococcus pneumoniae. Yeah. And so the next day we did real-time PCR for Streptococcus pneumoniae for all of them, and all of them were positive. Okay. Does it mean anything? Is anyone here a bacteriologist? Okay, or clinical settings, or well, no, that's important. This is not a question of molecular medicine or capacity. They are really positive. But the point is that this pneumonia could be present in the upper respiratory tract of people without any symptoms. Okay, so it even may colonize it, but without any symptoms, it may be part of not normal, but part of flora. So a molecular test is too sensitive for this. So if it is positive, it doesn't mean that it is the cause of the disease. Okay, but there was one clue in this setting, and it was that the CT, the threshold cycle for all samples were very low. And what does it mean, people from NIH? When the CT is very low in a real-time PCR test, there is a high viral load. High bacterial load in this case. So you were suspicious that this is not due to just contamination during the sample collection or during the sputum coming out of the nose and mouth. It should be real. But anyway, it is not conclusive. So what we did a PCR, a normal PCR for these samples, for these adenoviruses, and we tried to do a PCR for the bacteria, but we failed anyway. And for the adenoviruses, we succeeded in amplifying in 2 samples, and then succeeded in sequencing one. And that one was a type of 11A. And this was the strange point that this is one of the very few subtypes of adenovirus which causes pneumonia. It is very uncommon. What it was, there were reports of similar settings in Southeast Asia. So our conclusion was, this is a pneumococcal pneumonia in an uncommon adenoviral pneumonia background. And we had not access to the patient, so we did this analysis just based on the samples that we received. So this is an example of what we can do in the lab. And even if we really want that, we can do it in 48 hours. So it took 5 days for us, but we can do that. Well, COVID-19 was a more interesting story. Well, in January 2020, when the first sequences were reported, WHO released a guideline to how to test it. It was, I think, on January 11 or January 13. And their recommendation was doing real-time PCR for 3 parts of the genome, one for this part N, and one for E, and one for one AB. And also for this area, for this region, have true PCR to true specific parameters, because this PCR could be positive in case of SARS-CoV-1 2 in the original source. So they wanted to first detect it and then identify it or separate it from the original source in case that there is a problem. Well, it is very difficult to do 3 PCR, in fact 4 PCRs for one sample because we didn't also an internal control for that. Okay? It is not practical. So 1 week later, they changed the recommendation and just said 1 for teaching and 1 for wireframe B. And that was the primary approach they designed and circulated and delivered to WHO to different countries. Well, this was the guideline, the standard guideline. The standard guideline said a positive nucleic acid amplification test result for at least 2 different targets or 1 NAAT and followed by sequencing to make sure that this is SARS-CoV-2. Okay, this was the standard guideline. Well, the first day that we found the first case, the first day we tested 2 samples, the second day we have 40 samples, and in 3rd or 4th day we came to 400 samples, just in our lab. And it is really overwhelming. So what did we do? What was the problem? We did not have capacity to run 2 tube tests because according to this newer version of the recommendation, we needed 2 tests. We were not able to do 2 tests. We don't have enough PCR machines at that time. And our main concern was because our hospitals were full of people with this symptom. And the physicians wanted to know which are positive or which is not positive to separate them, just to focus on giving the treatment to the right people, and also avoiding the infection of the others. So our concern at that time was not to take mass screening. So we were addressing the requirement of people in the hospital. And the other problem that in one occasion, I just realized that almost 1/3 of all the samples that we receive is positive. So what's the point of doing a test? So if you pick up as one of the 33%, you have a chance to give the right answer. So what did we do? This is the genome. This is the genome structure of this virus. So it is an RNA virus, and when it is the genome, and these are the way that it is transferred to mRNAs inside the cell. And you see, the more you go to the end of the genome, the more copies of this part is present in the transcript. So when you have this transcript, it contains all other fragments, but this protein is the only protein which is produced by this transcript. And there is a gradient here, you see, and the last one is something like 30 to 100 times more expressed or more available than the original the genome. So what does it mean? It means that if you do the test on this region, what would be the difference of the result of this doing the PCR on this and on OR180. You have much more frequent template for this, because PCR on this gene is positive on this mRNA, and so on. Okay, so you have more template for doing the PCR. So your analytical sensitivity is higher. Okay, so it is more sensitive. This is why, even if you are an epidemiologist, if you are a medical doctor, you must know something about the biology of the target of the agent that you are dealing with. So we wondered what if we just test one gene, and we did this study. We tested a series of samples both for E gene and RDRP gene, and also for N gene, for the target. And then we did this analysis. This is, you see, this is here, and it is very well, the equation is very well fit on it. So, and you see all of them, the correlation is very good. All of them are positive, and some cases, you have, for example, in this case, E gene is positive, if CT is 30, but CT of higher CT is lower number of templates. So this is 31, but this is not negative. Okay, it does not mean that it's negative, but it's not detected because the sensitivity of this gene is higher than that. So we checked it again with the N gene and it was positive. Okay, so we thought why should we do two tests for this at this moment? And another point about this second test, as I told you here, based on this guideline, they were worried if there is another SARS-CoV-2 circulating in the SARS-CoV in the population. But because these are the family of SARS-CoV viruses, and they wanted to make sure this is SARS-CoV. But we knew that there is no other virus in the circulation. So whatever we get from one test means that it is SARS-CoV-2. So there is no need to do that. Okay, this was one calculation that we did. The second one is based on the epidemiological aspect. I told you who was of primary client at that time. It was not the public. It was the physician and the hospitals. We wanted to relieve the burden of the patient in the hospital. Okay. So what is important for them? The PPE. Okay. Well, you have a PCR test. It is very specific. It is very positive on a negative sample. But we know that we have some flaws in it. So because for that time we knew that some cases are positive, but we cannot take it. So based on this estimation, we did this calculation. If we have 100 patients, we can identify 75 of them, and we will miss 25 of them. But if we test normal people, and this is an overestimate for that, this is an underestimate, it's actually 100%. So, okay, 98 of them will be negative and 2 just false positive. Okay? But for finding these 100 people in the general population, you should consider the prevalence of the disease in the population. And in the hospitals people, I told you that this okay, and well, and if the prevalence is something like 4.1%, for finding 100 people, you have to test how many? 10,000? 1,000, sorry, 100. Okay, 10,000. 100,000, yeah, that's right, 100,000 people. Okay. So if you do the calculation, PPV is a probability of having a positive test in all, having the disease in people who have a positive test. Okay, this test is very specific, but in 100,000 people, if you test them, 2,000 of them will give you false negative, false positive. So the PPV is something like this. Okay. If you test it in a population with a prevalence of 25%, for finding, or 33% in fact, or no, okay, it doesn't matter really. For finding 100 people, you have to check only 400 people. So you have to have at least 300 healthy people, because the prevalence is very high. And in this case, your PPV is 75 divided by 75 plus 6, 81. And it is more than 9%. Okay, So based on these two arguments, we went to the National Committee of COVID-19 in early March 2020, and we convinced them that we do not need to follow the WHO guidelines. We just need one test at this point. And that was rolled out in the labs. Just 2 weeks later, this was the modification in the WHO guidelines. In areas where COVID-19 virus is widely spread, a simpler algorithm might be adopted in which, for example, screening by RT or RT-PCR or single discriminated target is considered sufficient. So you see, we were in the lab, but we helped the health authorities to make the right decision. We provided them the evidence that they needed to make the right decision. We suggested them to take the right decision. However, very shortly, we moved to testing for 2 years again, because at that time, multiple real-time PCR tests were available, and we were worried. Again, we were worried because we looked at the biology of the virus. We were worried about the mutation that which can nullify one test. So we did not want to rely only on one test. So we came back to the previous WHO guideline. Then another important point in lab service is you need to expand it. It depends on the prevalence of the disease, the extent of the outbreak that you're dealing with. But this is important. Well, we will talk about it, about how many tests you really need to do to control an outbreak in the control measures. But just to give you a view of what we did, our capacity, well, when the first case was reported, was it was 80 something like 200 tests in the whole country. And to show you the burden of what we did, you know, the most common disease which was tested in health setting of Iran before it was for influenza, and every year up to 25,000 tests were done for influenza in Iran. Okay. And in just about 1 month, we reached the capacity of doing 10,000 tests per day for COVID-19 in Iran. And our peak capacity was something like 100,000 tests per day. So you see how tremendous work you need to expand this lab. This lab of knowledge, you cannot just ask them to do this. You need training, you need logistics, you need providing kits, you need the standardization of them, you need quality control of them, you need many things. And we were, I think, I'm very proud of being part of this team. Well, one other question was raised in the previous workshop in Karachi was, what is your opinion about pooling the samples? When you have so many samples, why don't you pool them? So what is your opinion? Okay. Don't tell me that we should do this.
Because if you issue more than 10,000, 20,000 samples per day.
Okay. You know what pooling means? Means that you put the samples together, make a pool of them, do the test. If it's positive, then break down the pool again, do the testing again, find which one is responsible for the positive results. Okay. And this is what was done in China on several occasions. It was suggested from other authorities or other nations to do that. But finally, we didn't agree, or we didn't come to the conclusion that we needed. The point is this, based on this calculation. Consider that you have 100 samples and you prepare 10 pools of 10 on each. Okay. When you are going to find just one positive sample in these 100 tests, if you test them one by one, you need 100 tests, right? But if you pool them in pools of 10 samples, you do 10 tests, then one pool is positive, you break down the pool, 10 more tests, and finally you can screen the whole area, whole sample with 20 tests. Okay, it's reasonable. But when your prevalence is 3%, so this was 1%, 3%. So you have 10 pools, you test them, 3 of them turn positive, you have to break down the pools, you have to do 10 more tests. You see the increase of the number of the tests? And your primary step was 100 tests. If you are going to do 20 tests instead of 100 tests, that's okay. But if you reach to a level, for example, 5%, then you will get something like 60 tests per sample. And you know, this pooling is very demanding. People must very carefully, or the machine must mix the samples very carefully. Then you must store them, then you must come back to re-examine the food. Okay, so it is labor intensive work. Sometimes it's not really worth to do that. So based on especially based on the prevalence, you can decide whether it is helpful or not. And another point is that by mixing the sample, we decrease the analytical sensitivity of your test. So this is a trade-off. So it's up to you to decide if it is helpful or not. Or if you do it with tools of 5 samples per food. Again, in this case, we need 25 tests. In this case, 35 tests. So justify it. You do, people in the lab can discuss and sit and discuss with the people of the technology. Does it work to do that or not? Is it work to do that? Okay. And yesterday, Feynman showed you this kind of terms. Okay, these are the normal epidemics that is behind to the before the molecular medicine era, the traditional epidemiology. And well, in Denmark in 2006, after almost 20 years of having no measles case, suddenly they found out this pattern of measles. And if you compare this with these 4 times, what do you say? What kind of outbreak is this?
Common,
Intermittent. Okay, what else? Okay. Well, in both of them, you have a source who have entered the community and brings the disease to the community. But later on, they did a molecular analysis, and this is what they found. So these samples belong to at least 4 different clades of the virus. So what does it tell you? You have 4 separate outbreaks.
Yes.
Okay, and you can understand how the management can differ just based on this piece of information. Okay, and this is what we do for you. Okay. Well, this is true also for COVID-19. So if you look at this is either an intermittent or propagated curve during the time, right? But if you consider it more detailed, each peak is due to a different genotype. Okay, and we know in the case of COVID-19, the cross immunity is not very extensive. So it looks to me that for COVID, you can have maybe 4 or 6 different episodes of outbreak due to different strains. So this is not just one disease and one outbreak. And this takes us to the last subject of this session, which is genomic surveillance. And it is to identify, monitor the genomic variation of the disease, which is the focus. Usually we do it for 2 reasons. One is for epidemiologic monitoring of the pathogen, to see if the same as the example that I already gave you on measles and the other one, or even more complicated form to find out the source of the disease. So if you have complete genome sequence of cases in a period of time in different locations, different people, there are methods of analysis that Sana is the expert of that. To show you, to give you an idea what was the origin and when was the original introduction of this disease to this community. And this is also good for the environmental studies, which is the subject of this morning talk. So there are ways to find out what happened, but they are time-consuming and also very resource exhaustive. This is the original CRF 01A strain in Japan. And it goes back to 1980 something, 88, something like this. Or for case of COVID-19, you're familiar with this graph now. It shows that how it has evolved. And you see now, you cannot find Wuhan strain anymore in the circulation. But the others you can find. And you can trace this epidemiological pattern to some whatever conclusion that you want to draw out of it. But there are other aspects of that too. For SARS-CoV-2, after doing this analysis, we found out, I mean, not we, but the whole community, scientific community found out that the transmissibility of different strains are different. So that's important because it changes the course of the control measures that you have to put in place. This also can help for preventive therapeutic intervention. Again, for SARS-CoV-2, well, this is severity of outcome of the different strains. So based on this, well, we know that, for example, Delta was more severe than the others. So if you have this data on time, if the patient is of the older variant, maybe you can somewhat personalize the care that he or he needs. Decide what kind of care is more suitable for this person or for this community. You do not need to advise on what size fits all for the whole country or for the whole world. Or the other aspect was to monitor the immunogenicity or cross immunity between different strains. So if you check that, for example, for Beta variant, the vaccines against Alpha variant work well. So you do not need to change, modify your vaccination strategy. But if they are different, if you have a new variant like variants of Omicron, then you have to change it in a large extent. Well, and now these systems are very much available around the world. Thanks to COVID-19, many countries have good next generation sequencing, which provides the ability to test a large number of samples at once. So now the capacity is very high. I visited NIH in Pakistan, and it was amazing. You have several machines there. And this is what you really actually are doing. For COVID-19, you are doing it. For CCHF, you are doing it. For dengue, for rotavirus, poliovirus, and it's really helpful. It is a new concept which is now expanding in the community of epidemiology and also. Okay, that was my first slide. Well, I have 20 more minutes, and in the afternoon there was a little modification in the agenda based on, I mean, not based on, from what we did in Karachi. And for this afternoon, I need more than half an hour to talk about the control mission. So if you let me, if you agree, I can continue for maybe take rest for 5 minutes, maybe, or 2 minutes, and then start my presentation. Then we drop it. First in the afternoon, Anish will give you this lecture on fire safety, and then I will come back and continue this presentation. Okay? So just wake up, stand up. Stretch your leg. So just stop share. Okay. Now I open the
I have stopped the recording.
Yes.
Okay, please come back. Come back. Everybody, come back. That's it. Okay, come in. Please come in. One comment was raised by, with me. My phone is on. Was raised by, Secretary Strategy, I mentioned you again. As I told you, for calculation of PPV, you need to know the rebalance. And you cannot estimate rebalance based on case control studies. You need core studies, core type studies, the population-based studies. So context, not anyway, population-based studies are to get the rebalance. So you are expected to do the calculation based on the case control study. Okay, let me start just the introduction of my second talk this afternoon. It is about the control measures on outbreak. Well, to make you relieve my main conversation in the first slide, so what good movies? Okay, like what? Like this. This is an perfect movie of our country. I used to show it in my classes before COVID and discuss it with my students. And step by step, this is part of the, I mean, they follow what we really want to do. It is a case, an outbreak of an infectious disease, an airborne disease. It is, I think, from the SARS family also. And, well, unfortunately, the last, at the end, they beat me out of the stars. But not like this part, but anyway. What other aspects is exactly what you need to know. Have you watched it or not?
Sir, we have watched many zombies. Zombies, like they are the outbreak of viruses,
Yeah, no, but this one is different because World War Z, for example, is a similar concept, but that's a sci-fi. But this is, I heard that they have consultants from the WHO. And maybe some parts of it have been done inside the WHO. So it's very realistic. And also there is another outbreak. It is also,
Yes.
I do not like it also, but this one is different. This is really what a good movie is.
So outbreak is not included.
No,
Okay, yeah.
I do not like it.
And different
Yeah.
virus.
No, the point is that this is really textbook.
Okay.
Even before COVID, we discussed this movie with students and it's fantastic. So now we are done and go. Okay.
We can watch this on both sides.
On what?
On both sides.
On fourth day?
We can watch this movie.
When?
Yeah.
Yeah, in the afternoon. Well, yes, actually, if I have time, I would be very happy to have one session of watching this movie and discussing. But then we got it, short in time. Well, I showed you another version of this slide, which was given to me by Pierre Formetti in 2007. And it was based on the previous experience on Rift Valley fever and other outbreaks up to that time. This is very much similar to that, and I just wanted to tell you that the principles and the concepts are almost the same, even now after COVID. So the framework is almost like this, and you see you have clinical management, you have epidemiological investigation, logistics, behavioral and social interventions, and more important than anything here is coordination with these activities. In between, you have to see some critical aspects, you have media, and you have a lot of activities here that we will discuss about some of them in the coming minutes and years. And sometimes very tiny aspects of the culture of the people that you are working with is important, very important. Is in fact the most crucial part of controlling an outbreak for you. And we discussed this part of the risk communication and okay, I think it's the afternoon. Yes, risk analysis and risk communication and how to do the social mobilization. Well, of course, we do not have much time, so we'll just touch upon them, give you some examples. But this is the core concept of why we do the outbreak control. This is an example of Rift Valley fever, and the time scale is days. So this is a very rapid outbreak. And you have this disease in animals, then there is a spillover to human cases, then you have an epidemic, and this is the way that you do it. And well, when you detect, usually detect the first cases in the normal, in a system which is not concerned about early detection. Usually it is a late detection. So you detect them, the first human cases, you hear that people saying that there is a new disease, then some of them get here, some of them get to the hospital, some of them do the test, and then you realize that there's no outbreak. And you start your control measure somewhere here, okay, in the descending arm of the curve. And if you notice, this curve is not symmetrical. So, some part here, even before you do anything, the curve is somehow goes down faster than what you expect from normal curve. And that's because people, by themselves, by hunch, by whatever, talking to each other, and have some perception of what they can do to prevent to get ill. So they do something to help themselves, even if you do not do anything. And then in addition to that, all human outbreaks so far has vanished at some point. So if we do not do any intervention like what we did not, well, people did some intervention in the influenza of 1918. But even if you do not do anything, it infects some people, it kills some people, and then when enough people are immunized, then the disease is gone. SARS is an example. SARS, we do not care about SARS anymore. So you see that it is gone. What is important for us is to detect as early as possible. Then the time to action usually takes similar time. Now after COVID, is shorter time, hopefully, because we are more prepared now. And by the way, Dr. Ashaki said that today is the International Day of Preparedness. So that's why we are doing this. We are going to be prepared. So we do the interventions somewhere here, and then this is the opportunity of saving lives and saving health of people. So the concept is to detect the disease as soon as possible, or even before infecting people, because almost 70% of this emerging infections are zoonotic. And if you check the list of WHO for the threats, the biological threats of the community, all of them except for disease are viruses. This is local virus, but we don't know what it is. So, and they are zoonotic. So if you have a better health system, a one health approach, you can even detect them in animals. Or even better than that. Most of these diseases have some arthropod vectors, which are very sensitive to the climate. So, for example, there are mosquitoes. If you check the weather before that, if you do the weather forecasting or monitoring, remote sensing, but satellite things like this, you see the green coverage of the area. You see you monitor the amount of rain during the previous season, for example, things like this. You can predict that if you will have an increase in the vectors or not. Then you can be more prepared. So the whole concept is to find it as early as possible and do the intervention as soon as possible and do the proper intervention, of course. And this is our role. And another point, more complicated the picture right now, especially that was from Rift Valley, this is from Ebola. So now the importance of wildlife is more highlighted, and due to the climate change and urbanization, now we are in more close contact with the wildlife. So we are in more danger. So we have to put in place some system to monitor the wildlife. And if you can do that, it's excellent. It's very, very nice. Well, now let me start just with the epidemiology. Going back to this slide, you see that epidemiological investigation, surveillance, and laboratory are well different. But anyway, now we are in the phase that we have detected an outbreak, and we want to decide on what measures to put in place to control the outbreak. Okay, so we're not talking about just identifying the outbreak now, we are the time of controlling it. So when you look at this, well, you saw this kind of terms for. When you look at, for example, it's Hong Kong, this is somewhere in Africa. And you see the structure of the buildings. So these are the big area, the population density should be low relative to this. Here you have 20, 30 floor buildings with so many people, in so small area, encountering with each other every day. So you see that just looking at the place that they live gives you some clues that there are some differences between the epidemiology in this region. Or how many people one person can meet during the day, how he meets them, if people go, for example, to church, to masjid, regularly or not. All these aspects give you clues of how you can control the outbreak. And for Ebola, for example, principles of control, and also you must know the route of transmission, of course, and the vectors or the intermediate, components of this chain. For Ebola, it was control of transmission through case detection, case investigation, contact tracing, interruption of nosocomial transmission. This is the control of transmission. Safe and dignified burial. It is a big problem. And case management. What you should do with the people, with the patient, and with the people who take care of the patient. And all of them need your community engagement. So this is a very simplified form of the first slide, the big picture. Well, but let's talk about the rest, because we want to go more detail into the aspect in one area now. Okay. Any question so far? Thank you. Okay, shall we start? Okay. Guys, I'm going to discussion for 1 hour, 15 minutes. I'm going to talk about sorry. Oh, about biosafety and security. Really, the course could be around 1 week even for biosafety and biosecurity. And since I didn't know really the participants, who they are and how are their backgrounds, so I have provided a very compact basics of biosafety and biosecurity. Maybe it's a little bit no, maybe more. Very familiar for you. If it is just reviewing, it's worth it. And if it's new, I would be so happy. So it would be the basics of biosafety, biosecurity, fast and compact, because I have to end in 15 slides in around 50 minutes, 1 hour. So by definition, I will go to through the definition of the biohazards. What are they exactly? How they are classified? So then I will go through biosafety, the principles, the practices, and the biosecurity, and finally laboratory, good laboratory practices or good microbiological practices that are the main component of the biosafety and biosecurity. It would be the sequence of the subjects that I'm going to introduce. So, by definition, what is biological hazard? A biological hazard, then biohazard, is a potentially infectious agent or hazardous biological material that which
even agent or material, present a risk or potential risk to the health of humans, animals, plants, or the environment. So a general definition has. They could be found everywhere in the environment, soil, water, plants, animals, humans, and they are in their natural habitat, they are dangerous. When you come to handling them in their lab or genetically modifying them, you put a more dangerous or more danger or more risk to the environment. You should enhance their lethal properties or make them even resistant to preventives and therapeutics. So the definition may even goes worse this way. Here, types of biohazards you can see. They could be organisms, really microorganisms, bacteria, viruses, fungi, parasites. Biological active agents, toxins, allergens, venom. Certain types of recombinant DNA, RNA, they could be biological hazards, and other potentially biological agents. Means blood, body fluid, tissue samples, various cell line GMOs. If I'm going too fast, please tell me to calm down because I'm going to finish it in 1 hour. For most of you, they are very familiar, just for reviewing. As you saw, there are many types of biological hazards, biohazards, so a system for their classification has been introduced. And this classification is based on some characteristics. What are they? One of them, and the most important one, is the pathogenicity or virulence. The second, route of infection, mode of transmission, survival in the environment, infectious dose, availability of effective preventive therapeutic treatment, host range, natural distribution, impact of introduction into the environment. Among them, 3 of them are more important. And the main classification is based on these characteristics. I mean, pathogenicity, mode of transmission, and availability of effective treatment and preventions. Do you know the difference between route of infection and mode of transmission? You may know it. Okay, what is this? Briefly.
Particles are transmitted from one person to another, or how they are surviving environment.
Okay, and the other one?
Route is how directly it is infecting the person, and mode of transmission is a channelized process.
Yes, for gastroenteritis, for example.
Like many viruses are transmitted through oral fecal.
Yes, exactly. But the infection is in the gastrointestine, is inside the gastrointestine. Okay, that's good. Just to make you less sleepy because it's after lunch.
Yes.
I know that all of you know it. So based on these classifications, antibiological agents come under these 4 classifications: risk group 1, risk group 2, risk group 3, RT1, RT2, RT3, or RT4. You know them. Based on the individual posing risk for individuals and community, they are classified basically. For RG1, no or low individual and community risk. For RG2, moderate individual, low community. RG3, high individual, low community, and RG4, high individual, high community. And their characteristics, for RG1, they are unlikely to cause human or animal disease. They cause, but they are unlikely. For RG2, unlikely to be a serious hazard, but effective treatment and preventive measures still exists. For RG3, usually cause serious human and animal diseases, does not ordinarily spread from one person to another, and effective treatment and preventive measures are available. The difference with RG4 is that it can be readily transmitted from one individual to other, and usually effective treatment and prevention doesn't exist. You can find these information and even more in different places. The most credible one is pathogen safety data sheet. When you want to go to work with a pathogen, it's a very good idea to go for PS or to see its PSDS, we say, pathogen safety data sheets, to find out the control measures, the risks that it poses, and everything that is needed to know about that microorganism. And also, I recently found another reference, the approved list of biological agents. It's related to HSE of UK, and it has even more data about, more information about the pathogens that you maybe find them very helpful and informative. What is important for us as some lab workers and some field epidemiologists, as I was expecting, is lab-acquired infections. It's something like field-acquired infections for people who are going to work in the field for example, for taking samples or things like this. But the point is that we don't have field-acquired infections. It's something like lab-acquired infections comes under this umbrella. These lab-acquired infections are very important for us. Are infections whether symptomatic or asymptomatic acquired through laboratory or laboratory-related activities? It's like something like field epidemiology. There are mainly 5 possible ways based on the prevalence. One of the most prevalent one is spills and splashes onto skin and mucous membranes inside the laboratory. The second one is sharp injuries, the ingestion, inhalation of infectious aerosols, and bites or scratches from an infected animal. At least for me, by myself, it has happened. It has happened, and it has happened. Just ingestion and inhalation has not happened yet. I hope it happens very soon. Yeah. And the exact transmission route is identified in only 20% of LAIs. In 80% of LAIs, we don't know really what is the route of infection. If you work in the lab, at least one of them has happened to you so far.
It's a word-wise ratio. 20% is word-wise ratio. Yeah, it's specific to area.
Yes, they know that which of them has happened exactly.
Okay.
For 80%, you don't know what has happened. So there are involvement factors for LAIs that are very important for us as a lab people or field people. The host, the source, and the route. For the host, the susceptibility of the host is very important. Their immune system, how it works, they have susceptible immune system or strong immune systems, vaccination status, and age. All of them are important. Source of infection, is it microorganisms? Is it cells and tissues, body and body fluids, any items contaminated with the above? And route of infection. Percutaneous inoculation, it is the most difficult one to prevent. Inhalation of aerosols, contact of mucous membrane, and ingestion. All of them come in the close contact with each other, and lab-acquired infection comes out. There are some defects in the laboratories. I know many of you are aware of them, that results in the LAI or laboratory-acquired infections. Another point is that 80% of LAIs are caused by human errors, and 20% are caused by equipment failure. So training and having respectable, committed people is very important when you are talking about LAIs. It's different from those 20% that I told you that the route of infection is not clear. Those defects are mainly based on what has been released or published. Lack of knowledge of biological agents. You don't know really what you are working with exactly. Lack of proper emergency preparedness. Some exposure occurs, but you don't know how to handle it, to manage it. Lack of training, passing down of bad habits. Maybe you are like us, and this situation happens for you many more. And the last one, carelessness, ignorance in work practices. Do you have any experience that which one of them is more prevalent in your labs, if you are working in?
Last time.
Yeah, exactly. Exactly the same. Even for me, I was sometimes teaching it. Sometimes, especially when you are very tired. Yes, carelessness is the point, because you know that you should do, but when lots of experiments, lots of samples that are coming to the lab, and you get lost inside the samples. That's a place that carelessness happens. Among common agents that cause LAIs, 40% are caused by bacteria. Which bacteria are the most prevalent one? Shigella, Brucella, Salmonella, Staphylococcus aureus, and Neisseria meningitidis. Some of them are very dangerous, some less. And one important point, that if you are working with them, be aware of this. About 30 years ago, I was working in a laboratory for the first time, and we were trying to detect the bacterial cultures that based on the phenotypic issues through smelling them even. And Brucella was among them. And nothing about biosafety and biosecurity was present 30 years ago, at least for our laboratory. And one more point is that if you are working with microbacterial tuberculosis, you are exposed to 9 times more risk than community. It's a huge amount of risk that if people are working with M. tuberculosis, maybe you have the laboratory and you should be aware of it. So we are now aware of biological hazards, biohazards. We come to the biosafety and how we can control the biological hazards. By definition, biosafety is the combination of principles, technologies, practices, and measures. All of them, combination of them that are implemented for handling biohazard material that we talk about. The point is, biosafety is different from biosecurity in this place, that it protects personnel and environment from unintentional exposure. That's different from biosecurity, that you are preventing the exposure intentionally. Through biosafety, you contain the biological agents during manipulation and prevent environmental release. It's very classical definition and explanation of the biosafety. Now, how biosafety is achieved? At least 4 main parts or controls or procedures are needed for achieving biosafety. They are administrative controls, engineering controls, practices and procedures, and using PPE, personal protective equipment. About controls that should be posed by or introduced by the administration, they are training, issuing certificates, authorization of work, compliance verification, controlling impact transfers of biohazardous material, inventory control, and proper disposal and waste management. We will talk about some of them, not all of them. Engineering controls, they are commissioning and decommissioning of the lab, annual certification of BST, biological safety cabinets, autoclave safety, centrifuge and other equipment safety. Sonicators, homogenizers, workers. For people who are working in the lab, do you know what is the common characteristic among these equipment? I mean, centrifuge, sonicator, homogenizer, vortex, that poses risk for biosafety. Do you find any common characteristic among them that poses risks for biosafety?
Samples using centrifugation.
Yes, what happens?
Samples leakage.
Okay. And this is common among centrifuge, sonicator, homogenizer, vortex. It's common. It's say all of them has this risk. It's producing aerosols. Aerosols is produced by all of these practices using these equipments. And there are other practices and equipments that are also do that, because like freeze dryers. When you're pipetting the liquids inside the lab, they produce aerosols. We know them, they are inside the books, inside the textbooks of the biosafety. But sometimes there are practices that doesn't seem to produce danger or poses risk for the producing aerosols. Something like this. When you pull out the needle from a rubber capped container, as you see in this picture, lots of aerosols have been released. And that's why when you are doing such a practice, you should wrap the bottle with the absorbent, salt, and disinfectant. That way, if you are working with an infectious agent, and you pull it out, so you prevent the infectious aerosols to be produced. Practices and procedures, conducting risk assessment should be done. Very important component for biosafety. I will talk about it a little bit, and Dr. Kehan will talk about it more descriptively. Appropriate practices in appropriate containment. It's very important in which containment you are working with which pathogen. Submission of health assessment surveys and inventory. For PP, you are aware of, we will speak about them more in detail. in the coming slides: gloves, safety glasses, lab coats, closed shoes, and some other PPEs. I was going to ask them, but we are in time shortages. What's important in this slide is that we always say PPEs are very important. A characteristics lab member wears PPEs, at least lab coat, at least gloves. But when you go to control measures for biosafety, you see that PPE is the least effective one compared to elimination of the biohazard. Substitution of it with a less hazardous material, commissioning control, engineering controls, and administrative controls. If you have the option or say the chance to do some of them, you should do based on their priority. PPE, very important, but compared to these elements, it is the least effective one. So coming to biosecurity. I was in China for a biosafety course about 1 month ago. They have one word in China, in Chinese, that means biosecurity. They use it for both biosafety and biosecurity. They say that biosecurity is a more broad word, that biosafety comes under its umbrella. And it was very strange for me. It was the only country that's like this. And they have published a guideline, it's biosecurity, but biosafety is inside. You can't find something exactly means biosafety. They say biosecurity. And biosafety is already included. For biosecurity, the same principles, technologies, measure, and practices. It's like biosafety, employed for the protection, control, accountability of biological agents. It's not just biological agents. It's about data, equipment, biotechnologies, skills and information related to their handling. All of them comes under the definition of the biosecurity. That is used for unauthorized access, loss, theft, misuse, diversion, or release of each of them, or all of them. It's a very broad definition of the biosecurity. You can find different definitions, but it's the the broadest one, really. And now how it is achieved? Biosecurity, at least 4 items are coming here for biosecurity. You should use physical barriers, building exit control, locks, key card access, self-locking doors, you know them. The structural design that reflects the transition from general public to laboratory zones and designed to control traffic flow pattern and access. Both of them keeps your laboratory away from the ordinary or general public that are coming to your institution. Psychological barriers, very important. As you see for the army there, they use psychological barriers very more. Obvious presence of identifiable security personnel. It is a psychological barrier. Use of cameras, mirror, other monitoring tools, and overall awareness of personal, people, and behavior. Monitoring activities, patrol support staff technicians, key access, and personal clearance that you use keys, cards, and codes given to them. All of them are coming under the umbrella of biosecurity and how it is achieved. In biosafety and biosecurity, a very important subject that should be done even for every experiment you are doing is biorisk assessment. Based on that, based on biosafety, biorisk assessments, you design your biosafety and biosecurity measures. If you've done wrongly or in an incorrect way, your biosafety and biosecurity measures may not be effective. So a good biorisk assessment is needed, and many factors are involved in biorisk management. What are those factors are? Agent characteristics and biological material, personal supervising and personal using the material, environment, experimental protocols, and lab practices. All of them, when you are going to design an experiment or working in a place, all of them should be included in your risk assessments, in your bio-risk assessments. Dr. Kim would talk about it in more detail. But totally, there's a strategy. You should have a strategy for bio-risk assessment. You describe your work and activities, identify hazards, it's goes step by step, determine the risks, decide whether the risk is acceptable or not. If yes, you should go for your work, you proceed for your work. If no, you should remove or close the project. Or if a risk control action plan can be used, you should implement control measure and review your adequacy of plan. If it is going to be makes risk acceptable, so you go this way and you can do your experiments. By the way, doing it personally, I mean, by yourself is very difficult job, a very tough job really, because we used to do that and it took a long time, and finally you find some factors that you have forgotten to include. Nowadays, there are some websites and models that help a lot in this way. They are based on mathematical models, but you put some data inside the website and then you get the risks that have been prioritized for you. It's very good that even it has been prioritized. The properties of particular biological agents, lab security practices, in place mitigation measures, and likelihood of the malicious use of the biological agent are included. So one of the most famous one is a bio assessment model. We call it BioRAMS. You go to the website. You can go for risk assessment, laboratory biosafety, laboratory biosecurity, and see how risk assessment is done and how the risks are prioritized for you to make your measures to control the hazards. It's very helpful. As I told you, based on the risk assessments, you should go for the containment of the biohazards. You are going to contain it. You are going to limit it, the exposure of the biological agent. Based on the containments, classification, there are two kinds of containments: primary containments and secondary containments. What are they? Primary containments are first line of this defense, and they ensure protection of personnel and immediate environment from exposure. Here is the difference between primary and secondary one. The secondary one, you ensure protection the environment outside the lab. It's relatively further. This primary containment is achieved through good microbiological practices that we talk about in more detail, and use of safety equipment that we talk about also as well. As an example, protective envelope that encapsulates the infectious agents or animals like petri dish, vials, biological safety cabinet, and animal caging equipment are primary containments. And the secondary containments, what are they? They are going to protect environment external to the laboratory. They could be directional airflow, air drain and filtration, HEPA filter of lab air, pressure differential, laboratory design, operational practices. They are not directly things that we are using in the lab, but they are secondary containments. Now, based on the level of containment, primary or secondary, and which of the containments have been included in the lab, either based on the engineering, operational, technical, and physical requirements, the containments are classified into 4 groups again, by containment level 1, level 2, level 3, and level 4. One point here is that we have RG1, RG2, RG3, and RG4 previously. It's not the case that you should work with risk group 1 pathogen in BCL4 exactly. It's not like this. It's based on your risk assessment. It's based on the activity that you are going to do with your RT4, for example. If you are going to detect it, just to detect, for example, by molecular methods, it's a BSL-2 plus or BSL-3 is enough. You don't need. But if you are going to culture an RT4 pathogen, you should go for a BCL4, biocontainment level 4 laboratory. So you should find that you should manage. You could manage to balance them. Which RT1, which risk groups go to which biocontainment level laboratory. And increasing the number of BCL increases the potential rate, the procedural and facility requirement, the level of operational containment, and the degree of protection for personnel. So BCL4 is the most demanding one. And many countries are trying to avoid having BCL4 because it's very expensive to maintain it. And the point is that if you have BCL4 or BSL4 laboratory, you should have something valuable to do inside it because it's very expensive to maintain. So your work should worth to work in it. The Chinese were telling us that if you want to have a BSL4, try to find something worthy at first, because you pose a risk to the community when you have a BSL4 that you don't know really what you are what to do inside it. Maybe you go in a wrong way, something like those gain of action experiments that pose a risk to the community. So, as I told you, a very important part of the biosafety is good microbiological practices, as we talked about it. What are they? That is a general practice that all types of work involving microorganisms, irrespective of containment level, should be done through good microbiological practices. It has some items that we are going to talk about it. It prevents both the personnel, the workers of the laboratory, and also contamination of the experiment and samples also. I mean, both of them are being protected from contamination. These are some items of the good microbiological practices. GMP, you know, we have a good manufacturing practices that's for production, different things. But these items are included, but not limited to what? What are the items? Use of PPE, uncluttered bench space and disinfection of work surface, using the aseptic technique, minimizing use of aerosols, proper use of BSCs, spill management, hand washing, identification and preparation of biological waste. We will talk about most of them, a little bit, of course, for each of them. So we go to start with PPE, because it's very important. Good microbiological practices is very important in the lab. What does they do? What do they do? They provide a barrier to protect the worker from potential exposure to hazard. They just protect the worker, not the sample itself, as we expected from the good microbiological practices. But does not remove the hazard nor guarantee complete protection for workers. So you should be aware of the limitations. Some other points for using PPE is coming. When is PPE necessary? What PPE is necessary? Which PPE you should use? What is the limitations of the PPE that you are using? How to properly wear PPE, because it's another important point that many people don't comply with it. How to care, maintenance, and storage of PPE. And here, this is me, working in the laboratory in 2015, I think, for Ebola detection. We had some suspected cases. Fortunately, they were negative, but we should be prepared. So we had the mobile lab inside, and we were working with the samples that were coming. As you see, this is the almost everything that we had in the lab I have put on. There is nothing left. guns, cameras, 2 gloves, boots, shoe covers, scarf, face shield, goggles, everything. I have a goggle, also a medical goggle, also medical glass also inside too. And it was really hard to work inside such a lots of food because I was afraid of, and we didn't know really. It was not a guideline that for Ebola detection you should wear this or that you shouldn't wear this. We had someone from WHO coming to see our lab how we are working, and you said that you don't need to put on like this. It was warm, and I sweat a lot normally. And I was sweating in a way that my sweat was accumulating inside my shoes even. And it kills really.
So we have faced the COVID-19. We wear face shield,
Yeah,
mask,
I see.
So it's too much hard to.
Yes, exactly. After 2 hours, you see the world is changing. Yeah. And it's really not needed. So you have a good plan for using PPE. Based on what I said, there are some principles to choose your PPE. What are they? You should have your risk assessment. Your PPE should match the hazard. If you use a larger group that is low, RG group 1, you don't need to put on lots of PPE. Based on the transmission route and activities. What secondary containments exist inside the lab? And comfort and convenience. It's very important. Maybe you have to use, but I have an experience working with the glove boxes. We had a company in our country that was producing glove boxes, but because it couldn't provide gloves for biological ones, it had installed chemical resistant gloves. So they are very difficult to flex. Even me with these fingers couldn't, let alone the women that we are trying to work with. So convenience and comfort really matter sometimes. Common PPEs, what are they? Lab coats and gown. How should they be worn? Must always be worn closed, never wash with regular clothing, and never wear lab coats in offices. I am sure that you have the same problem. You have people that are wearing coats in office, conference rooms, lounge rooms outside. For gloves, there are kinds of gloves: latex, nitrile, rubber, vinyl. Be aware that some of them are not proper to work with viruses. Pore sizes are different and should not allow them be used with working with viruses. Very important, washing hands after removing the gloves. They should be disposed in a proper container. They should not be worn out of the lab. Or even you have the same problem, people that are working, then their phone rings and they respond while they are wearing the gloves. So it's a very bad practice. If a glove fails, you should consider it like an exposure, and you should go for washing it and seeking for medical attention. Eye protection, you all are aware of that safety glasses, safety goggles, face shields that mostly should be used along with either safety goggles or safety glasses. You can hear that how a medical glass, a laboratory glass, a goggle, and a face shield could be effective in preventing a large splash on the face of this maquette, for example. You see that goggles and glasses are not effective at all. At least goggles have protected the eyes, but not the other parts. And in the laboratory, really try to not using the contact lens, because if something happens, it poses a large danger and more complicated the process of washing and managing the situation. And proper hand washing is important too. About masks and footwear, I'm going to talk about in more detail because they are very important. And about shoes, maybe you have the same problems. People are using shoes that are not proper. Do you know which the shoes that you are using in the lab have, how they should be? Closed toes, closed heels. Closed heels should be closed toes and closed heels.
So it's full life.
You know, then what's the point? If something really spills on your food, you will have a very hard time to rinse it, to wash it. It's very different from the hands. And when you are in stress and you are alone, finding some way, even some place that you can wash your shoes or your feet is very critical. And you should be aware of that using right shoes is very important inside lab because we had such an experience. One more point about PPEs is the sequence of PPEs. I'm not going to explain it all of them. You can download it from the CDs in a 3-page PDF file. They're very informative and very brief. But when you are going to put on regular PPEs in the lab, you should go putting on through the sequence of first gun, second mask, third goggles, and finally gloves. When you go to remove them, this is the process of donning and doffing. There are two options. You should go from the gloves to goggles, guns, masks, finally, or even another option is that gun and gloves at the same time, then goggles, and finally mask. Both of them ends with washing your hands. But this, for Ebola, there were lots of PPEs to be worn, and we had a long list of one person was standing and the other was putting on. And the same was going for doffing because we were very careful about the virus because, as you know, the mortality rate is more than 60, sometimes 90%. So based on your risk assessment, you should be very careful about the PPEs and the practices that you are using your PPEs. Now about masks, because I found this table recently because I had many debates about the people in the coronavirus pandemic were using surgical masks or say single-use face masks. I found this table from a company that will produce a company website that were producing masks. They had it has divided the masks to single-use face masks, surgical masks that I was expecting that they are the same, but they are not, and respirator mask. For surgical face mask and or single-use face mask and surgical one, as you see, they are more than 90% effective in filtration of 3 micron particles, and not effective for particles that are about 0.5 microns. For surgical mask of China, with this code, it's made in China. I think the company was Chinese too. For 0.1 particles, microparticles, the effectiveness was more than 30%. And a very strange one was this USA ASTM F2100 that is exactly like an N95 mask. For 3 micron particles, it's more than 90%, and for 0.1 micron particles, it's even more than 95% effective in protection. It's somehow different, strange. I could not find a supportive data for it. I don't know really if it's written correctly or not. That's why I told you that I found it from the website of the company. So it doesn't fit really a surgical mask with N95. What is the difference? But for the European code, it's normal. It's what we are expecting. For 3 micron, it's good. For 0.1 micron, no protection. And for N95, that is very important. That is very common. You know that how the N95 and FFPs are interrelated. It's based on the classification of USA and Europe. If you find mask that is N95. It's an American-based classification that is equal to FFP1 in European classification. If it is FFP2, it's N99. If it's FFP3, it's N100. And it's based on that all 3-micron particles effectiveness of 95%, 99%, and 99.95% effectiveness in filtering of the particles. So these masks are good for splashes and large particles. And the point for N95, 7, 9, and 100, you should be aware they do not protect against chemical hazard. Because I have seen people that they are working with chemicals, they are using N95, and they think that you should use chemical masks. No way. And you should do fit test if you are working with a dangerous pathogen that is airborne. A fit test is necessary for N95. Here I found a data from the CDC. I think it might be interesting. No mask, no protection. It is for SARS-CoV-2, for COVID-19. Even for cloth mask, 56% effectiveness has been detected. It's good. That's why I always say, blaming people that why you are using cloth mask. This more than 50% is effective. Surgical mask, 66%. Even not much more. Maybe we had to be more economic and use cloth mask those days. I didn't know it. And for N95, it is 83% of protection. Because splashes was very common way of transmission of the SARS-CoV-2. If a virus practices in a different way, maybe cloth or surgical masks may not be effective like this. Okay. Other good laboratory practices that are important to reduce or minimize the exposure, especially for people who are working in the lab, are when you are going to open tubes, you should do it when you are working with an infectious agent. I mean, all of them, they are when you are working with a biohazard material. The opening the tubes should be done in biological safety cabinet. When you are going to open, first unscrew the cap a little bit, stand for a few seconds, and then you can screw it completely. It is very important, especially when you're working with the liquid nitrogen tanks. If you are working with it, we had the problem inside. One of the ladies in our laboratory, student, brought up the cryovials, and it exploded. It explodes. No way. Sometimes maybe you have not seen, but you may see in the future, because the atmosphere inside gets combusted and makes these plastic cryovials like splashed everywhere, and it cut her face even, because it was near. We were so lucky that it didn't hit her eyes. Yes, if it was a disaster. When you bring out the cryovirus from the liquid nitrogen, firstly and immediately, unscrew it a little bit, stand, and then go to the BSC to unscrew it completely. It's very important when you are working with the liquid nitrogen. For pipetting, discharging the pipettes should be in a place that is very near the level of the fluid that you are working with it, and do not forcefully expand the pipette. It's very important to prevent the aerosol formation. For syringes, I told you, avoid whenever possible, because it is the most difficult one to prevent from the risks that it poses. You can't wear the gloves that are resistant for puncture. It is very difficult to work with. And withdrawing with the absorbent that I told you, and do not bend, shear, or recap radius. That is completely clear. For centrifugation, for biological material, for biological hazards, there are some special centrifuges. They have caps on it, and they don't, if they even produce aerosols, they are not released outside. You can do your centrifugation stand for a few minutes. All the aerosols settles down, and then you can uncap your rotors or say your tubes. And you should be aware of their O-rings. There are the lasting parts that keep it airtight. And use of flasks, you should try to not using glassware, just plastic ware. It's very much more safe. And when you are using microtubes, I mean, don't use snap caps, use screw caps when you are working with the biological material, because when you use snap caps, they readily produce aerosols when you open it. Hand hygiene, I told you, very important, good microbiological practice. It should be done frequently before starting any manipulation. Very important in this situation before leaving the lab. Whenever the integrity of gloves is questioned, before and after completing any task in the BSc, every time gloves are removed, before contact with face or mouth, and at the end of the day, all of them. You should do your hand washing really frequently. It helps a lot in preventing the infections. I will tell you an example that's a published article at the end, of course. Another component of good laboratory practices is waste management. You sure will have waste in your laboratory that you should be aware of how to manage them. Based on the characteristics of the waste that is produced and associated risk that is posed by the contaminants, I mean the biological agents here. Two means of the waste disinfection, say decontamination, I mean, are available mostly in the labs. One is the chemical agents, using chemical agents that are mostly bleach and ethanol. I think that you are mostly using bleach too. Bleach and also. And physical means, autoclaving and incineration. Do you know which way, physical or chemical, are more effective?
I think physical.
The more effective, irrespective of the material, I mean. Physical is more effective. Physical is more, because really temperature, high temperature, and incineration destroys everything. But for chemicals, you will see there are many limitations to use them. If you have the option to use either chemical or physical, to be sure, use physical ones. There's a definition for decontamination. Decontamination is the process of removing or inactivating infectious material, toxin, microorganisms to a level that removes danger of infection to individual. This is my definition. And it's divided to two classifications, two parts, either disinfection or sterilization. Disinfection eliminates most forms of the living organisms, while sterilization, complete destruction of all viable microorganisms. So in sterilization, you actually destroy all the microorganisms that may exist. So it's a more advanced kind of decontamination. For chemical disinfection, chemical disinfectants mostly, sorry, yes?
In sterilization, use temperature or some kind of chemicals to sterilize things.
There are some chemicals that you can use for sterilization. But let's go to the end of this slide and see how possible that the chemical disinfectant result in a sterilization, because it's a tough job. Generally, disinfection with chemical disinfectants mostly are kinds of disinfection rather than sterilization. You will see how it is done. Most common ones are chlorine compounds and alcohols. Choices of chemical disinfectants depends on type of material to be disinfected, organic load, chemical characteristics. You know the main disadvantage of using bleaches in the laboratory? What is it? Oh, it's killing. It's a disadvantage still. If you, because as you know, the pH of the concentrated bleach is 12. It should go to the around 6 to 7 to release its chlorine ions. So that way it is disinfects. It disinfects. When you go for acids, mixing with acids, some people use it for cleaning some places, that has killed someone even. You go for pH 4. The chlorine ions that are supposed to be released in 1 hour or 2 hours are released at once, and it is toxic for people. That's why it shouldn't be in contact with acidic solutions. But the main disadvantage, based on what we are doing in our lab, is the inactivation of the bleach when it comes in contact with organic material, including blood. And the blood deactivates the bleach very readily, very easily. So you have, after using a little bit of blood to be disinfected, you should change your new solution of the bleach to be used again. And every day, every 24 hours, you should have a new working solution of the bleach from the concentrated one. You can't keep your bleach solution for about 1 week or 1 month. One day, it's done. You should change and have a new solution of the bleach. And most of them has this disadvantage that they should penetrate. They should come in contact with inside the materials that they are going to be contacted. And assuring such a phenomenon, such a happening, is not as effective as using temperature, high temperature, I mean. That's why using chemicals for sterilization is very risky. And also, the contact time is very important. As you know, for bleach and for the alcohol, the same. You should give them enough time to have their effect on the biological waste. The most used physical method for the contamination, here sterilization, is autoclaving. But the major point is, for some items you can, and for some items you can't use autoclaving. Mostly you know which items can be autoclaved, but those that cannot be autoclaved are chemicals that have flammable oxidizer, phenol, acid, or alkalides. They are not proper to be decontaminated or sterilized by autoclaving. Chemotropic or radioactive waste, very important, and also bleach itself. And because it releases lots of chlorine ion that are very toxic to the environment. Another point in good microbiological practice. What time it is really? Okay. Yeah, maybe in 10 minutes or less. Another one, emergency preparedness and response. Very important. You should have your risk assessments before to foresee and mitigate the emergencies. You should have a pre-plan or SOPs protocol to how you forefront or confront with the emergency situation. Be familiar with the emergency spill procedure. Telling them and doing them really is different. We see them here, we hear them from different people, but when we go in practice, we see that how many shortages we have in the lab. I'm sure that you have the same as we have. Be familiar with emergency escape routes, locations of fire extinguishers, IWASH stations, safety showers, and first aid kits. This is my personal experience. We had I was in a laboratory that something happened for my eye, and I was alone. The eye wash machine was really complicated. With something in my eye, I was trying to find it how it works, and it was really complicated and a very hard time I had. So be aware that in your laboratory where you work, how equipments that are related to biosafety are working. Really important. Sometimes it happens and there's no one to help you. And the last one is carefully planning your experiments based on the biosafety guidelines to understand to save your life, really. These are the emergencies that happen. Whatever you do, for sure they will happen. Mostly they are not limited to, but mostly they are splashes that aerosol generated, spills inside a centrifuge, and spills inside a biosafety cabinet. What happened for my eye is that I was trying to break a little bit of glass in a glass pipette, and when it broke, it came. I have glasses, and it came from the side of from the beneath of my glasses and went into my eye. Very unlucky, but it happens.
If you are not very well in glasses.
Yes, yes. Those glasses don't help at all, for nothing. Even I hope it never happens, but in car crashes, they are very dangerous. And they cause the blindness even. In case of exposure, you do everything and exposure happens. What you should do, immediately and safely stop any current task and safely remove PPE. You are in an emergency and you should go to remove PPE. It's very important to not pose yourself in a risk during removing PPE, because PPEs might be infected with the microorganisms, and when you are removing it, you pose a new risk for you. So in those stressful situations, you should be aware of how to remove PPEs or have someone to help you. Immediately wash the affected site with the soap and water and soap. Water with 1% soap in it, liquid soap, is one of the best washing solutions in the laboratory. It washes almost, it's very cheap to provide and very available and very effective. You can even wash your glassware in the lab with the 1% soap and water. For mucous membranes, including conjunctiva, you should wash it with a copious amount of water or eye wash solution, and you should go for informing your supervisor and finding or seeking infectious disease expert. Spills happen. Again, spill of biological material. I'm not going to talk about chemical spills, but they happen more frequently also. But spill management should be done immediately after the spill. It's a very good idea to have a spill kit inside your lab. It's not something complicated. It has very cheap and attainable components inside. Concentrated disinfectant, spray bottle, paper towels, autoclave bag or biohazard bag, forceps to pick up broken glass, sharp container, and PPE. So it's not very complicated. You can have a spill kit inside your laboratory. To manage your spills. How they are possible? You cover the spill with a paper towel. You pour disinfectant on it, for example, 10% bleach. And you should work from outside to inside or outward to inward, and leave it to 20 to 30 minutes to be sure that the agent has been inactivated. So you should make sure no traffic around the spill and keep the ventilation on if the spill happens inside the BSC. In case now spill inside the centrifuge, you should keep it closed and let it for around 30 minutes for the aerosols to go down, to settle down, and then you open it and decontaminated in this way. Last point is the transfer of biological material. It should be based on some standards. There are some codes, Dangerous Goods Act or acts, I mean. You should have proper classification, proper packaging, proper labeling, proper documentation, and import-export permits if you are going to send or receive biological material from a foreign country. You may now be aware of it. You should have your biological material in triple package container. First one mostly is a plastic container only because it's resistant to be broken, contrary to glasswares. The second one is a watertight packaging that is a rigid one. And the third one is a rigid outer packaging. So a third layer packaging is a standard one. But when it goes to be, things go crazy, like what happened for SARS-CoV-2, we had many samples from different parts of the country coming in different way, leaked, broken, and you couldn't have the chance to say that, oh no, we don't accept the sample because it is not in the standard mode. That's why, based on the risk assessments, somehow risk assessments go psychologically. You should decide which package you should accept. As an example, if you have a very dangerous biological material that is coming to you and it's not very prevalent, so you should have these regulations to keep people and yourself safe. That depends on the situation.
If you remember, once we rejected the samples from Parliament, the same reason.
Yeah, those days the things has been a little bit settled down, I think. Yeah, once from the Parliament they sent samples that were packaged not in a standard way, so we had the brave to send it back. I'm not sure how we could. Here is for the importance of hand washing. I'm going to tell you. This is published in 2016. This is a biological technician, laboratory technician in Turkey, microbiology laboratory. He was infected with Staphylococcus aureus. They go for identification and for the PFGE to find out the exact characteristics. And they found that it is a very exactly the same that they had inside their freezer from another patient. They couldn't find that how it is possible that he has those. I told you that just 20% of the route of infection are clear. So they came to find out how he has been infected. They saw that the doffing practice of the gloves of this guy was incorrect, and the very bad practice was that not washing his hands when he was leaving the lab. So they could find out that just with washing his hands, he could prevent the Staphylococcus aureus infection in the lab. All in all, collectively, all of these stakeholders should be participate in the biosafety and biosecurity, lab regulation, technical standards, design construction, risk assessment, and personal training, along with the emergency response. You should find more information if you wish. This is Laboratory Biosafety Manual of the WHO, the most valid one, BMBL of CDC and NIH. These global guidance framework for the responsible use of the life science published by WHO, Laboratory Biosecurity guidance by WHO, and you yourself in Pakistan have National Laboratory Biosafety and Biosecurity Policy. It's a brief guideline, 36 pages, but it's very informative. And you will find it, if you haven't not seen it, it would be good for, especially for people who are working in the laboratory. So finally, I think you think maybe all of us think safety, act safely, and be safe. And I hope you safety always. Thank you. Any question? Fingers.
May after and of course share with us the scientific PowerPoint.
Second point.
Share with us the scientific PowerPoint.
Yes, you're here.
After
Yes.
the
Okay. Thank you. Sorry to be so fast and compact. No way. Yes.
Why we are not using sanitizer after washing hands?
You can use it. You can use it. Sanitizers, alcohol, these sanitizers are along with, it was in the slide, but I have to quickly. You can either use non-washing by soap and water, or you can use, alcohol with sanitizer. No, that's not you can use. Yeah, that's right. But I prefer one because it rains.
Yeah.
If it is, because alcohol evaporates and the percentage comes down. If it is not working, it won't. Maybe mixing them is more effective.
Yeah.
But yes, thank you. Yes.
So I have a question. So as you described in your presentation, different categories of masses, and they are like they have potential to remove the particle at different proportions. So sir, you are talking about with serum which are we are like what about the air? Is there protection involved?
Yes, because you know there are some medical department masks that was very prevalent put on the body that was exhausted. Maybe you have seen it. Different exhausted. They were not more brilliant in COVID-19 period because they said they don't filtrate the outcoming that's why the outcoming rate is filtered to the mass. Because the people that are infected should be able to do complete mass without filters.
Actually, I was thinking about it. If a person is infected with a contagious virus, they can spread through the air.
Yes.
And they use mask, like they.
Because the mechanism of that mass is that these 5 layers of chain cloth. One of them in sometimes in carbonated ones, active carbonated. This is going inside or outside, they don't do anything special on the air that is going and coming back. So when you expand the air, they don't, they are not in the place that it can inhibit. That can, no, they're unable to filter the air that going down. Okay, thank you.
Just. Okay.
Feedback, so, I'm sure you some, people from you will make later. I thought that is interesting enough. How is the oh, yes, that's. Okay. Well, Bismillah ar-Rahman ar-Rahim, we reached at this point that the aim of the outbreak control is to reduce basic reproduction number to below one. What does it mean? The epidemiologists. What? What is population? So what is R not? R not, or R zero? Yes? What is it?
Zero.
What?
To control the outbreak is zero.
No. It should be just below one. And what is R zero in itself? What? No, formation you mean? No.
The spread of disease.
Spread of disease, okay, so what is it?
It tells the spread length of the disease, how it is emerging.
Well, not really. R naught is the number of in this very easy way, is the number of people who can be infected from one infected person. Okay, but there are some minor differences between different types of R. In general, R, E, or E dot or zero, and that this number is considered among people who are at risk, who are vulnerable, or all general population. Anyway, doesn't matter right now. The point is that how many people can be infected by one person. So if you are infected and you transmit it only to 1%, not more, so you have a serious state of disease in your population. This is the definition. We expect the number of cases directly generated by one case in a population where all individuals are susceptible to the infection. So its estimation depends on the duration of infectivity, the longer it is, more people are infected, the likelihood of transmission, then contact between the susceptible person and someone else. So in any encounter, if the chance of transmitting disease is higher for the disease, so it's more contagious than the other one. And finally, the contact rate. How many people do you meet during this activity period? And if it is more than one, means that every 1% infects more than 1%. So you have a growing between. If it is one, so it's stable. And if it is less than one, means that every people from, for example, if you have 2 infected people in a ward, we have only one. So it is decreasing, goes to zero. And the aim of the aim of this all these control measures is to reduce the R1 to below zero. In that case, it could be controlled. Well, again, going back to Ebola example. You're talking about the epidemiologic investigation during the outbreak, okay? So we must be able to find anybody who is infected during the control. So by finding him, we can trace contacts, we can try to isolate him, we can try to cure him if it's possible, and then monitor the contacts for the whole period of incubation time, the maximum incubation time. So if they're not infected, we release them. If they are infected,
We can diagnose them very quickly and then do the control measures on them. Well, contact tracing is very important. It is an active surveillance to break transmission chain by detection of new cases. So if you check everybody around someone who's infected, find it, you can find the cases as soon as possible. So the duration of transmission is decreased. Because after that, you can isolate, you can do something to not to transmit from the second guy to another guy. Okay, so the duration is decreased, so you can control the outbreak. And the question is that who is the contact? This is very dependent on the type of disease and the type of transmission and things like this. For example, in Ebola, what was the route of transmission? It was not airborne. It was not. It was based on droplets. Okay, so airborne is smaller, the particles are smaller, and this is not airborne, it's not droplets. So you must be in really close proximity of the patient, and the droplets tend to drop on you. So the definition was has slept in the same household as a case, has had direct physical contact with the case, either alive or dead. That's also important. For example, maybe for COVID, I'm not sure, but for COVID, the dead people are not so dangerous because they do not breathe. But for Ebola, it's important. And and this definition includes healthcare workers, so the nurses or the doctors who are treating them, has touched the blood or body fluid, including urine, feces, vomit, tears, or sweat of the case during the illness, has touched the clothes or liners of the case, a baby who has been breastfed by the case. So these are the definition of contact in case of EBD. But well, as I told you, this may differ and differs really for different diseases. So how long should you take care of them, and how many of them? Well, in this case, it was considered to be 10. I think this is the normal number for this kind of activity in epidemiology. So if I'm right, you may correct me. But we, even in COVID, we consider this number in the beginning. And the number of contacts depends on the time between appearance of symptoms and isolation. So the longer this incubation time is, the more people you must consider. And for an effective control, you need to control 80% of contacts. So in that case, you will be able to reduce the break chain of transmission, reduce the R0 or R0 of the disease. Well, for Ebola patient, every contact should be visited every day. Well, by visit, it doesn't mean that physical visit. In Ebola, you could even call them, ask them if they have fever, if they have any symptoms or not, or ask someone else to check it. No need to taking body temperature by a thermometer. And contact tracers should keep at least 2 meters between him and the contact. And there is no need for PPE. So why this 2 meter? Why should people on this sample 2 meter with this patient or with the contact, which is not the patient in fact? Because the droplets cannot move more than 2 meters. Okay, and this was the case for COVID-19 in the beginning too. And there is no need for PPE. So the risk is okay. Hi, welcome.
Yes, sir. So if incubation period is more than 21 days, so in these cases, so what is the scenario of, so you should be that after 20 days, 21 days, or it is possible or not? So
You should continue.
No, it's okay. If more
Okay.
than
If the incubation period is more than 25 days was the maximum time for a COVID for a Ebola patient to either survive or die.
okay.
So that's the maximum. So if your incubation time is more than that, so the whole period of incubation time was monitored. Okay. And of course, you need ongoing epidemiological monitoring, and also studies during the outbreak. These studies are very important. I will talk about them a little bit. And you must identify real correlation. You know, these studies are important because many solution comes out of people. They say that doing this is good, doing this is bad, and you must collect evidence during the outbreak to see if they are right or not. Or if you have a guess that this measure can control the disease, you must check if you have done really or not. So that's why, again, this kind of studies that Pioneer mentioned yesterday are very critical. You should do the case control or core studies during the process of the audit. But one thing is important that we are looking for causal relationship between something and the outcome, which is the disease. Okay, so if people say that you can wash your hand with vinegar for COVID, does it work or not? So you need the evidence. And these are the criteria developed by Sir Austin Bradford, and these are called Bradford Hill, sorry. And these are called the criteria of Bradford Hill. So how do you consider a relationship to be causal? What are your criteria?
Variable 1 happened before variable B.
Okay.
About time.
That one point is time. Okay, what else? What was the environment? So what are the one?
It shows the sensitivity for the cause.
You might be somehow right, but your phrasing is not good. It's so.
Covariance about the variable 1.
Okay, so if you modify, if you change the first one, second one is changed, the outcome is changed. If you decrease, it's a decrease. That's all, or increase of the question. Okay, so what else? So these are the criteria. The strength, consistency, specificity, temporality, biological gradient, plausibility, coherence, experiment, and analogy. So you look for this kind of relationship between two factors to be able to say that this is, doing this measure can control the outbreak or can worsen it. And what are they? The strengths, well, you are looking for something that really works. So if you do something, but the effect is very low, okay, it might be statistically significant, but it's not worth the fact to try. So strength is one factor. Every time that you see the cause, you must see the effect, or at least most of the time. The more, if yes, you see cause, but there is no effect, so this relationship is not really causal. It should be specific. So one cause, or one cause results in one outcome or effect, and vice versa. So the ideal form is this one-to-one homology between these two. Then the time mentioned, biological gradient. The more you introduce the cause, you must have the more you must see the effect. Otherwise, it is not really a causal effect. Well, there are some exceptions when there is a threshold to see the effect of the cause. Below that threshold, whatever you increase the amount of cause, you do not see the effect. But after that, you see it. Then it should be plausible. You should be able to explain some theory on how it works. It should be coherent with other evidence, for example, from laboratory, from anywhere else in the way. And sometimes, if it is very good, if you can do the experiment, if you can, all these experiment challenges, I don't know. Clinical trials, for example, are much better than the observation that you collect during a case control study. So the type of study is important also, and you have some similar explanation in other cases or in other diseases, for example. During COVID, there was a large dispute, or not dispute, in fact, large number of theories about what drug works really, and most of them are rejected. So even and some remdesivir has very low margin of failure. So these kind of activities are done during the outbreak. Now we come to social mobilization. I'm going to talk about this also in the risk communication later, so I'm not going to discuss it in more detail here. But you see that you must know people who are influential in your community. Okay, this is the voodoo magician in Africa. So if you need to have, if you need their help, go to them and ask their help. To convince them to help you in a way, finally, you want to see the effect on the health of people. So it doesn't matter who is going to do that. Or we will discuss about that. Then you have to control the vector or environment, especially in the case of vector-borne diseases. And it is very common. You know that the life cycle of mosquitoes, for example, for malaria, for any disease transmitted by mosquitoes. Controlling the environment to prevent their reproduction is critical. This is the way dengue, for example, was transmitted to several places through water in the tires. They can put eggs there, they replicate, or you see the below this what is this? This parts of flower. I don't know. Okay. What will you say? Can you see what it is? So sometimes these are really important for the operating studies. Okay. No, not this. Okay, so it comes to case management. Well, it's critical. If you cannot do anything for the patient, so why are you isolating them? Why are you diagnosing them? And when you diagnose them, the site of the hospital is a very important source of transmission of the disease. So you must do whatever is needed to control the disease transmission there. This includes the plan of the area. So you maybe have to divide the area to different areas. You have some for susceptible cases, some for sort of probable cases, and some for confirmed cases. And also maybe some support for the covered patients. So this physical isolation and you need PPE for caregivers, for doctors, for nurses, and these are very important. In fact, this IPC, infection prevention and control measures, are very important during an outbreak. And again, they differ from disease to disease based on the route of transmission and the susceptibility of people. And another important aspect is the burial of the victims. And as you know, for Ebola, it was a big problem. For CCHA, it used to be a big problem in Iran because, as I said, even after death, these are transmissible. So, and it is our practice, it is our religion that we must watch the death before burying them. So in Iran, for CCHF, we came to the conclusion, or we received this fatwa, that we do not need to watch them, just bury them. And just to show you, this was the training for Ebola, and just for burial, we had 12 steps. So from donning and doffing, from preparing the team, donning and doffing, I don't know, wrapping finding the case, wrapping it instead of kafan, you should have something to just wrap them. Then transferring them from the site to the cemetery. Some places people have this, they want to be in the same car as the dead body, so you must convince them not to do that. There's a lot of job, and finally burying them and removing your PPE. So these are really complicated in some cases. Then it comes to, again, my favorite part, the lab. Okay, so well, we talked about how lab can help during the outbreak investigation. Now we're talking about control of the outbreak. So for case finding or for providing, you must disseminate the service in the area. Sometimes it is possible to collect sample, transfer them to the lab. But sometimes you have to take the lab to the site. So maybe in some cases, this is our own lab. This is the lab that we prepared. It's a molecular biology lab, and when we open it, It's a real-time PCR, a very small laptop to control it, and then you have kids inside it, and they take it to the periphery and then open the box and then install the system. Or I think I showed this video. Yes. Okay. This is another view of the same. This is our mobile lab that we prepare for Ebola. You see the structure. And you can transfer this to nearby the epidemic site and do the test, all the tests here. Well, I think Arash mentioned that this is over PPE. In fact, at that time, didn't know what. Now with COVID, we were much more relaxed. But anyway, sometimes it's necessary. And another important point that which test suits you. And the answer depends on what parameters do you want to test. We talked about surrogate markers and also direct markers for finding the direct evidence of the pathogen. And also the cost of molecular, this just to tell you the story of in the beginning of January, in March, February 2020, there was a lot of pressure on all governments, including also us, to find a measure to reduce the need for lockdown, to screen people and let them go to work. And at that time, several people came to us to say that we can produce rapid tests or we have rapid tests. And we were under a huge pressure at that time. And then we started to study. So what kind of antigen, what is the target antigen for COVID, for example? There are several antigens that could be used as a target, and different kind of antibodies also. Well, according to our previous data, we know that usually IgM is the first antibody which goes on, then IgG. Okay. Well, and there were ELISA tests, they were developed at that time, and then rapid tests. And at the same time, the British government bought pre-ordered, in fact, 3.5 million rapid antibody tests, and they claim that now we have solved all the problems. Don't worry, we will deliver it to the household, so you can test yourself. If you are negative, you can go to work. But very soon it became a scandal in the British government because of this. This is another paper published in April 2020. And the scientific world knew that this might be a problem. In fact, we collected some samples from the patient in the first month, and when we received these fields, we started to test. Sensitivity was about 80% hospitalization. Okay, and what was the purpose? The so-called purpose of this test to screen normal people, not patients. And in the patient, we had this sensitivity of 80%. Like this was very low. The specificity was more than 90% because we had samples from the past. We tested them and they were negative. And we asked, why are we going to use this and what is the purpose? Then some other papers came out, and this was very interesting because it was published in the month, I think it was in June 2020. And they checked almost 28 people, 26 people in fact, and checked the IgG and IgM of them every day. And what they found was interesting. Okay, 7 people, based on our classic understanding, develop IgM, then IgG. Okay, but 9 of them develop IgG and IgM at the same time. And more interestingly, most of them, 10 patients, had IgG seroconversion before IgM seroconversion. So that was very interesting. So in case of COVID-19, in case of in fact, coronaviruses, sometimes even you have IgG before IgM. But the other point is that this meet the seroconversion day, something like 13 days, 12 days. And by 12 days, at that time, we knew that the symptoms have been full blown. So there's no need for testing. So we found that there's no need for this kind of test. So we stood and we prevented importing any of these cases and even going any of these cases to the national public health services. And that was a very good choice.
So what is the meaning by the scenario of the like self. Yes, no.
It's
Yes, so how to
think-
define this? Actually, IgM and IgG at the same time, so how?
No, these are synchronous. They use different kits
Okay.
to check for IgG and IgM. So in this case, we are very proud that we didn't buy it, we didn't go for that. Well, another important question, how many tests should be done? I told you this, I showed you this graph before. Our capacity in the day zero or in the first day that we take the first case was something like 200 tests per day in the whole country. And we went up to 100,000 tests per day. Was it enough or not? Okay. Doctor, why not?
I think you are talking about the COVID screening, COVID test. During the peak of that pandemic, I think 1,000, 2,000, that is a very, very good amount.
Okay.
For people.
I'm talking about 100,000 tests per day. Just to give you a reference, for example, for comparison, Germany has the same almost population as Iran, almost 80 million people. And their capacity, the peak capacity was 1 million tests per day. And just to give you another reference, in China, they screened the whole cities of as big as, for example, Wuhan, just 10 days, with more than 11 million people. What? 16 million? Okay, now it is 16. Yes, come back from them very fresh. Okay, so my point is that what is the maximum? What is the needed number of testing? Well, what is the purpose of testing? Okay, I told you how we changed our strategy in the beginning. So we were going to respond only to clinics where patients, inpatients. Now we are talking about the process of controlling the outbreak. So we're talking about asymptomatic people or all patients. And in this case, well, as a rule of thumb, usually if you have about 10% of your tests are positive, that's okay. Because below it means that you're over testing. You test too many people. And above that, it means that you are under testing. So you are missing some people. And in some occasions, in some peaks, we reached up to 60% of tests were positive. So at least as a policymaker, we knew that the actual number of people patients are more than what we report. But it doesn't matter because anyway, everybody has symptoms, so you cannot do a specific measure to control it. But for the capacity, this rough estimate is the good index. Well, then the logistic that I told you. I showed you a photo of my colleagues in the lab, and they were happy this morning, if you remember. And that's very important because you see, you have a country to deliver your kids. So, do you have roads all around the country? Can you deliver by road? Or do you need the airports? Or if you have big rivers or small rivers, then you have boats. And even for the death, for the burial. Do you need coffins? Do you need the Japan, whatever you need? Well, just to tell you another interesting story, this is a system developed by Russians, and it is composed of 5 or 6 trucks, and one of these is the BSN-2 class lab, so they can do many things, many diagnostic tests. Even with agents of class 3 or even class 4, you can do the diagnostic process in class 2 or BSN-2 class. Well, they use this thing to control to monitor the health situation during the Sochi Winter Olympic in 2014, and it worked. And really, I'm not a pro Russian in this case, but anyway, I admire what they did. And each of these teams had something like 35 people, staff anyway. And Russia was very eager to send it as a tool to the biological people convention as a tool to monitor the outbreaks and things like this. And also they deployed one of these systems in Guinea during the Ebola outbreak, and it worked. Then once I asked one of these authorities that, okay, just tell me what is the system that is very good for all countries. And I asked him, could you please tell me how much vodka did you deliver to during the weekend to Guinea? He started thinking for a few seconds, said, a lot. And then he got my point. You cannot deliver this system in this Islamic country because you cannot support your people. So you see, this logistic is not just about kids, it's not about equipment. It is about, everything related to mission. All requirements. So even the vodka. Well, all measures that I talked about are, basically targeting two aspects of the outcome. Either, of the outcome. Either they target the etiology, they try to control the source of infection, the pathogen, or they try to control the transmission. And in many cases, not in many cases, but as Arish mentioned this morning, we are still not, SARS, the source of SARS was taken 15 years after its outbreak. And for SARS-CoV-2, we do not know exactly what happened. So sometimes we really do not need to know what is the cause. But even if you see that caution these days causes this, so it's very easy, just we need access to this case, to this test. Okay, so the measure, doing the measures, putting the measures in place does not really need to have an extensive knowledge of what's going on. Just see what's happening and then decide based on that. And for example, okay, for intervention efforts directed at source, for example, quarantine exposed persons. Okay, this is very common. This is also common practice in many countries. But you have another type of quarantine, for example, when you try to protect susceptible people, and this is called reverse quarantine. And this is what the people in Denmark and in what is that the region was called, Scandinavian people countries, they did. So they made sure that all people are isolated from the others, but they didn't shut down. When the country was not shut down, not just isolated this. So you see, but different measure, if you get the same result, it doesn't matter. Okay, if the cat gets the most, it doesn't matter black or white. So let it. Well, I have these two slides. I will discuss them later, and yeah, I still have a few minutes if you let me, please, because we have a guest today to explain for us what your experience in Pakistan. I would like to just briefly touch the concept of risk management. Well, of course, first of all, risk assessment and then risk management. Well, and also risk communication, which we'll leave probably this communication for tomorrow. Okay. Well, you remember this picture from the first day of COVID-19. Okay, these are streets in China, and they are sanitizing the street with this foam and this, what is it called? Not foam. Foam makers, yes, exactly. Okay. I didn't know. Was it really necessary to do that? How? I think not at all. Okay, because at that time you were not, they didn't know what is the risk.
What is the cause of?
What is the cause, what is the risk? And but when we saw this picture, we thought that this is more serious than what China is saying. So otherwise, they did not have to do this management of the risk, this kind of management. So it showed that their perception of risk was more than what they claimed. Or this is in Iran. People started to sanitize the ATM with liquid, sanitizers, and they destroyed a lot of them. So was it necessary? So you see this assessment of risk is very critical in everything that you do every day. Even when you do your daily activity, even if you want to take a test to pass the exam. Okay, what is the risk of failure? What is the, I mean, is it too big? Is it a final term, for example, the exam? Okay, it's different from a midterm exam, for example, in this case. What if you, for example, I don't know, whatever you do during your daily activity, so you do a risk assessment, and based on your assessment, you do a match. Okay. You know Anthony Fauci, right? Anthony Fauci? Okay, Anthony Fauci is a very big figure. He's actually the program of all physicians around the world. Because when I was studying medicine in 1994, 1995, we used to learn the internal medicine based on textbook of Harrison's textbook of internal medicine, which he was editor in some of edition and editor-in-chief in some of specific edition. So he is very known figure in the world of medicine. And he was the chief advisor for outbreak control in the United States. Okay, so that's one thing. You know Mark Ronnie? Okay. It is because do not watch good movies as I mentioned. He is an American comedian, an Iranian American comedian, and he is a very funny guy.
A black one?
Who?
A black one?
No, it's not that. It's false. Just I have a short clip of him,
Yeah.
and I hope that this works. Well, so far so good.
Every time I think this thing is over, Dr. Fauci shows up. And every time you think it's over, you're flipping the channels, Dr. Fauci. Okay, what are you gonna do? Even if you get a shot, you're gonna get two shots. Get 2 shots. Get 3 shots. Get 3 shots. Get 4 masks. 4 masks. 5 masks. Get 5 masks. 4 shots. 4 shots. 5 masks. 6 feet. 12 feet. 18 feet. Try turning your back. Turn your back to the guy. Don't look at the guy in the eyes. Do not look the guy in the eyes. You could get COVID eye. COVID eye is very bad. You don't want COVID eye. The Delta variant has COVID eye. You could get the Delta variant. You get the JetBlue variant. You don't want the Spirit variant. The Spirit variant is a really bad variant. It's very low budget. Do not get the Spirit variant. Is he mob? He's mob, right? Dr.
Okay. My point is this: the title of this slide. If you cannot make good decisions, okay, people will mock you, even if you are Anthony Fauci.
Yeah.
And during the outbreak, this is very critical. So be very, try to be as accurate as you can. And okay, this happens anyway. So of course, Dr. Fauci didn't give his advisors in the out of blue. So he had some evidence, and based on those evidence, his team did a risk assessment, and then they communicated the risk with the others. So that's the point. So what is risk? Risk is a combination of the probability of occurrence of a harm. So you have something harmful, but what is the probability of it to happen? So in the most simplest way is the multiplication of the effect of harm by the chance of happening. But it's usually more complicated. Mathematically, it's a function of severity and probability. So if you think that the outcome is very dangerous, even if the chance of it happening is very low, you try to avoid it. But if the probability, for example, is higher and the outcome is not very big deal, so maybe you decide not to do that. So the way that you deal with this risk depends on your perception or your assessment of the risk. And for the, let me see how this slide can go on. I will stop at this slide. So I have. So risk assessment. in general, the process that you identify the hazard, you analyze and evaluate the risk with that hazard, so the probability of its occurrence, and then you determine appropriate ways to eliminate, mitigate or control the hazard. So you cannot always eliminate it. Sometimes you have to control it. And I will give you tomorrow perhaps a very good example of that what I found in Pakistan. USD. That's very interesting. So, as I mentioned, what is the risk of failing an exam? What is the risk of failing an experiment? But that's also important. When you do experiment in the lab, when you have a small amount of genetic material and you want to clone it. Okay, before doing the cloning, you usually start doing the same process with another gene that you have in hand in a large quantity, so to check if all the steps are working or not. Then you go for the precious material. So this is a way to assess the risk and to mitigate the risk, and many other things. So the question related to what we are talking about is what is the risk of not taking a specific measure to control an outbreak? And that's what I want to talk about it with you tomorrow. Okay. Okay. Doctor, do you have slides? Okay. Could you please help us with the? Thank you for coming.
अगर आप कहते हैं तो मैं इस पे शेयर मुझे आई हुई है
अच्छा जी, अस्सलाम वालेकुम and very good afternoon. I can say good evening to all of you. I'm pleased and I'm glad that I'm here in front of you. I'm much more thankful to the Iranian colleagues, public health specialists, and comstech University for giving me the opportunity to share some of the past experiences of COVID-19. COVID-19. CCHF and as well as dengue, we had responded at national level. So few of the colleagues are from my institute, and also few of I know some of the colleagues as well. So my name is Dr. Shafiqur Rahman, and I am the scientific officer at NIH since 2018, and I am the FETP advanced course graduate that is being certified from US CDC, and also I have done a master's in public health. I am the course coordinator for frontline FETP training. So we are in introduction. We go back in the very brief introduction about the what is FETP, and so. It's not working. So sorry. No. So Pardon me if I go into the our national language Urdu. Apologies in advance. So have you heard about FETB?
Yes.
Okay. So what exactly? Okay. So what is this training program is about?
Very much application of the epidemiology, and I think so MBBS veterinarians are most so more 2 years.
So basically, this program, FETP training program, was initiated by US CDC. Are you people aware of US CDC?
Yes, sir.
Definitely. This was initiated in 1975 by US CDC, and currently in more than 65 countries, this program is running, funded partially, completely, or independently of the US CDC. So again, the Pakistan was lucky that in 2005, after keeping in view the core capacities of IHR International Health Regulations, most of you people might know कि हमने उसको हमने जब फुलफिल करना था तो हमें हमें कैपेसिटी वर्कफोर्स ऐसी चाहिए थी जो कि स्किल्ड हो, knowledgeable हो regarding the surveillance, regarding the epidemiology. So the NIH, under the supervision of Ministry of National Health Services, Regulation and Coordination, US CDC, and TEFNET started the FETP program. So, Alhamdulillah, till date, more than 390 graduates have been trained across the country from different organizations, institutes of the Pakistan, working in the health department, veterinary department, and different other departments in order to have a skilled workforce in the field of public health. So it offers on-job training for all health professionals across different institutions and organizations, advanced course. It comprises of 2 years of training, 4 weeks followed by 6 months in their duty stations. The milestones are being given to them, and after 6 months, they come again for classes and they update their milestones. So while the frontline training offers 3 months short course on epidemiology, surveillance, and data quality. So more than 300 graduates in advanced course and more than 400 the healthcare professionals are being trained in frontline training. So let's come to the dengue fever or the dengue outbreak we had faced in 2019. So what is dengue fever? Please. Yes.
They are kind of disease transmitted by mosquitoes.
Okay.
Okay.
Yes.
And it causes fever, fever, and staining bones, and many symptoms are there, and it's deadly.
Deadly. Why we call it a deadly disease?
Because
Dengue hemorrhagic fever. If the person develops the symptoms, about 15%, 15 to 20% of the patients go into the dengue hemorrhagic fever, and then if left untreated, the fatality is more than 90%. Okay, that is why we need to take early steps in order to control this disease. So, as you mentioned, that the dengue fever is transmitted by mosquitoes, Aedes aegypti and Aedes albopictus. Are these freshwater mosquitoes or fresh? Okay.
Clean water.
Okay, what is the difference between the fresh water and the other stagnant water?
Contaminated water which is stored in dams, tires,
Okay.
and they are stored in.
So what is the risk for us if it is the fresh water?
We have
in
to,
that What is the risk?
we have to make this water safe.
What is the risk? What is the risk?
Because dengue.
हमारे घरों में किस तरह से वो रिस्क है कि जी डेंगू के हवाले से
रोज कंटेनर नहीं होते जो हमारे वाटर होती हैं वो रोज कंटेनर जिसकी वजह से डेंगू के लिए एक फेवरेबल एनवायरनमेंट।
हमारे घरों में क्या कंटेनर हो सकते हैं?
पौधों के रिश्ते इन्होंने दिखाए कि इसमें पौधों के गमले होते हैं, बारिश का पानी अक्सर हम छतों पे या वैसे ही स्टोरेज हमारा स्टोर होते हैं उसमें हम बर्तन या इस तरह की चीजें टूटी फूटी वो रख देते हैं, बारिश हुई पानी वहाँ पे जमा हो। इस तरह की छोटी मोटी चीजें जो हम नॉर्मली अवॉइड करें नोट भी नहीं करते सारी सोच सकते।
Exactly. So the disease burden status of dengue fever changed significantly in Europe and American regions, and now they are also facing regular dengue fever outbreak. For the last 30 years, Pakistan is endemic to dengue fever. Do you know when was the very first case of dengue fever reported in Pakistan? Please. Sorry, 2006.
I
1978 was HIV. 1976 it was. Okay, so 1982 from the province Punjab. Now you just observe the trend of the disease. Also recorded 1994, 95, 97, and most cases recorded from Karachi. 2006 reported from north of the Pakistan, and approximately 5,800 cases were reported from across the country. Okay. Now the largest outbreak, it was in 2011, followed by, you can see, in 2015, it was KP, most affected province. Then 2017, 18, and 2019. In 2019, it was a major outbreak which affected the whole of the country. Previously, as we had discussed that the cases were sporadic or the clusters were being reported from different regions. But in 2019, it was a major outbreak which was reported from most of the urban areas of the country. So, on 31st August 2019, print media reported 200 dengue fever cases in Karachi, followed by 2nd September in Peshawar. So, our division, Field Epidemiology and Disease Surveillance Division, currently renamed as the Center for Disease Control, under the leadership of Ministry, activated Emergency Operation Center. Have you heard before the Emergency Operation Center? Emergency Operation Center. This is a new term for you people. But in US, in other developed and developing countries, Major public health disasters are being catered, responded with incident command and management system. Okay, and that type of leadership or that type of command system is being run in the emergency operation centers. Okay, so I think HSC से कितने colleagues हैं? HSA. You must be aware of NUC. कभी सुना है आप लोगों ने NUC का? आज तो मैं कोर का किताब भी देख के आए NUC का. What is that NUC? National Emergency Operations Center. What was its role? Its role was to integrate different departments which are concerned while responding to public health emergencies. Okay, so in case of dengue fever, what do you think? Which stakeholders, departments, institutions, or organizations need to be engaged? Sorry?
Public health, like primary healthcare centers,
Primary healthcare centers, we can categorize it as hospitals, okay. Sorry?
CSOs should be taken important, like community. Civil society.
Okay, civil society organizations, okay.
5-star doctors. We have to improve our 5-star skills, doctor,
Okay, so healthcare workers working in the hospitals. Keep in view the dengue outbreak.
Educating the message is also important for that.
Keeping in view the dengue outbreak, what do you think? Which stakeholders you need to engage? This is what emergency operation center works. This is how emergency operation center works. Okay, all stakeholders, not all stakeholders, relevant stakeholders. So, you can see the provincial health department, district health offices, vector departments, vector surveillance, okay, law enforcement agencies. What was the role of law enforcement agencies in controlling or in responding to dengue fever? आपने कभी देखा है पिंडी में लोगों का चालान होते हुए? It was after 2011. If there is a container found, यही चीज, किसी के घर में अगर डेंगू का लार्वा पकड़ा गया तो उसको जुर्माना होता है. So we had to taken or do the law enforcement agencies, vector department, by vector department in order to contain the vectors. Okay. Also, we need to engage civil societies like the Pakistan Red Crescent Society. Also,
I'm sorry. Then we have 10 more minutes.
yes, yes, I know. So, sanitation department, we also engage sanitation department in order to contain all the hot spots for dengue. So all the coordinated activities were incorporated in daily situation reports, and this was widely distributed among all stakeholders through emails, and also the data was displayed on the dashboards of Public Health Emergency Operations Center. See, so the STREP stand is the main reporting tool to monitor response activities, initiated by rapid response teams at the national and sub-national level. So in case of CCHF, is we all know that recently in Balochistan we had a outbreak of Crimean Congo hemorrhagic fever. Why it was erupted or much more highlighted? Do you know?
I think so. It's a new type. So it's also not.
As I mentioned earlier, that the first case of CCHF was reported in 1974.
No. This outbreak is different.
Why? This is my question. Why this outbreak was different from other outbreaks?
It was more severe.
In Balochistan, it's always more than 40% the case fatality of CCHF.
Pakistan specifically.
Yeah, that's
With the
virus is different. Yeah, this
It
time
was a
outbreak
reason.
is
It
different.
was a reason.
evidence.
No. The healthcare workers were developing symptoms of flu-like illness as well as thrombocytopenia.
So you're seeing like every time in Pakistan specifically, the fatality rate was more than 40%, right? And can you give some examples in previous examples?
You can,
CCHF.
yes, you can search out the data. You can search out the articles because why Balochistan, I'm not blaming the healthcare system, no. Because the cases report to the hospitals, visit to the hospitals very late, in the late stages. That is why Balochistan encounters more case fatality rate as compared to other provinces.
Social acceptability is the reason.
But
There are other
outbreak
reasons
from
as well.
Karachi. Recently, AQ has tracked a lot of cases that
Yes.
originate from Rajasthan.
Basically, yes, you're right. So, the provincial disease surveillance and response unit was reported that the healthcare professionals working in the medicine ward were complaining of flu-like illness and thrombocytopenia. So, the team was composed with these objectives, and the following FETP alumni and fellows were engaged to control, to respond to that outbreak. So I think few of the fellows you might know from this list. So these are the fellows from the Balochistan Provincial Disease Surveillance and Response Units. Later on, the team of lab personnel and these epidemiologists from NIH was also deputed to provide the technical support to the provincial team.
So your team has not considered veterinarian, if I am wrong. So why you do not consider your veterinarian? Because it's related to. Why your team does not contain veterinarians any, if anyone, I think so, I don't know. They are dropped coordination. Veterinarian rice make?
You can see here.
Okay.
It was, the team was composed based on One Health and multidisciplinary. Okay, so more than 330 people were interviewed and were sampled. Out of them, 20 came positive. So index case was also identified, who was from the district machine and dairy farm, who was a dairy farm worker. First on 17th October, he was admitted to the ward and then shifted to the, on 21st October, he was shifted to the isolation ward. Then this was the index case which erupted the outbreak. So, as One Health perspective, the 21 blood samples were collected from animals, 65 ticks were collected, and also the CCHF virus was isolated from animal sample and as well as from ticks. So 180 environmental samples were collected, out of which 6 came positive for CCHF, including ambubate, these machine, dustbin, aspiration unit. So, also, apart from that, the epidemiological or field investigation labs also proved that the virus was similar. The genomic sequencing was done, and it showed that the virus was same it was circulating in previous outbreaks. So we have also a FETP trained the person who is leading the Department of Virology at NIH, and according to his findings, the virus belonged to the clade IVA of the Asia-1 genotype. And this is the same which was being also isolated in 2011, 15, 17, and 19 cases.
Why the symptoms were not the same?
Sorry, all the patients had similar symptoms.
No, this sign, similar has not, this is some more modification. Yeah, this is confirmed and because.
Hello. We tracked all the cases, all the healthcare workers, pharmacists, security guard. The symptoms were similar to the typical classical symptoms of CCHA. But as the healthcare workers were involved, that is why the fatality could, alhamdulillah, it was not as we were expecting from the past experiences. So the Pakistan FETP's team's rigorous and innovative strategies, from infection IPC to the deployment of RRT to the high caliber of public health interventions and commitment to responsible communication, meticulous data collection, and rigorous scientific research, and poised to contribute the high impact journal, reflect the program dedication to excellence and continuous learning. So these are the fellows, these are the frontliners who work day and night in this outbreak. So this is Dr. Abad. He is the lead from the PDSRE Balochistan and his team. And it was a high-level meeting chaired by Secretary DG Balochistan. So this was
It is good.
It's good, keeping you the time limitation. Just overview of FETP and dengue outbreak. So if you have any question.
Sir, I have one question. Basically, I'm also from Gujarat, that's why I'm going to ask a question. So why should not they have their own, faculty? Why they are hiring faculty always when they do handle some kind of sudden outbreak of infectious diseases? They actually the hospital and other influences have to respond to contact. Why actually there is no facility and there is no faculty to control this outbreak?
You are saying that in responding to those outbreaks, why we do not have local experts?
Yeah, why? As I said, they are FETP, yeah. So
in my very first slide, I stated that the FETP program trains those healthcare professionals working in their departments,
In the respective areas.
in their respective districts or provinces.
have you a plan for to train all the health workers and your own provinces? You have, I'm talking about it. Have you a broad plan that how to counter this infectious disease each province, or you have just only a one center inspector?
No, as I mentioned earlier, that the NIH is the Federal Disease Surveillance and Response Units. which is leading to the surveillance and outbreak activities. But in provinces, there is Provincial Disease Surveillance and Response Unit, which is autonomous. And under that leadership of PDSRU, Provincial Disease and Response Unit, there are DDSRUs, District Disease Surveillance and Response Units, okay, which are running parallel, dependently or independently of the PDSRU. So there is a team that, although we know that is the world's fifth most populous country with limited resources, limited economy, limited constraints, we have not much skilled HR in field of public health, but we are glad that if we have few, they are highly skilled. And it's proven from this outbreak that this outbreak was conducted by Balochistan team. This outbreak was conducted solely under their leadership. We just provided the laboratory support for genomic surveillance. Otherwise, they have their Sentinel public health laboratories as well. Okay. Yes, please. Okay, the question. This is the question which is asked by the Nadrasha, that what is the geographic extent of dengue fever outbreak in Pakistan? So I have already mentioned in my presentation that Pakistan is faced unusual trend of outbreaks across the country. We had initially in Lahore, followed by Peshawar, then again in 2019 we had at national level, then 2022 2023 we had much more cases, more than 91,000 cases from across the country. So but good thing for that outbreak was that the case fatality rate was very low as compared to the other previous outbreaks.
What is the rate of CFR?
So it was less than 1%. Yes, less than 1%. Yes. So again, another question. Yes, did you isolate the virus with culture? Definitely, the virus was also isolated and genomic sequencing was done as well. No, through molecular assessment.
Not in the culture, right?
It doesn't mean. Yes. Yes. I'm not confirmed with this. Yes, Masab can answer it very well. So how did you suspect to CCHF? Yes, specific definition. Definitely, for any outbreak investigation, we definitely develop a suspected case definition, probable and confirmed case definition. The team developed a suspected case definition for those
to whom they were investigating. So I think it's done. Okay, I know it was a very short time, but we could discuss much more on these topics. Yes, sure. Definitely, we are the